Thank you, Vasilis, and thanks to all the Yale staff and faculty who have organized this, Dr. Johnson and others. Much appreciated. You know, the concept of exposome is something you're going to be exploring this afternoon. And in this day and age of omics, one can wonder, how does this all fit together? And that will be, I think, a recurring theme throughout the day. The Yale School of Public Health has had distinction in environmental health sciences, but I feel like we're on a rebuild and an ascendancy in that discipline at the present time. We got a little too small, and that you need a critical mass in this discipline. So with university support, we've marshaled some resources so that Vasilis and his colleagues can build in this area and other areas that make sense for our Yale competitive advantage in this discipline. And I just wanted to let you know that as a brand new dean, I have been here 9.5 weeks. So to say that I don't know what I'm doing would be an understatement. However, I will, oh, that's on the internet now. But you know, speaking frankly, happily in my career, I've, on a number of occasions, as an infectious disease epidemiologist, I've dovetailed into the world of environmental health sciences, have found it fascinating at every turn. I do teaching overseas. I teach on water and sanitation. I'm kind of an EHS wannabe. And I think that will be in the self-interest of your department, because I'm very ambitious on your behalf. I really want to see Yale be as effective as we can possibly be within our campus, with our engineering school, with our forestry and environmental studies, with some of the scientists in the medical school and in faculty of arts and sciences elsewhere. I want to see us networking with our regional partner schools when that makes sense to be more competitive. And I really don't want any fences or boundaries to constrain the kinds of partnerships we're looking for. Modern science doesn't do well in a silo. And the whole concept of the exposome sort of implies that I'm right on that, that in fact we're going to need a highly interdisciplinary approach to really tackle how environmental exposures interact with the rest of the omics, the genomics, the metabolomics, and on and on. So without further ado, I welcome you to Yale School of Public Health and look forward to a great symposium. And I'm happy that even though I've got a busy day, I'll be rejoining the symposium a little later in the afternoon. So thanks.
So we'll play the little video clip by, oops, what did I do wrong?
Hello, everyone. I'm Dr. Linda Birnbaum, Director of the National Institute of Environmental Health Sciences. Many of you know us as the NIEHS. I want to thank Dr. Vasiliou and the Yale University School of Public Health for hosting this very important symposium and for inviting us to contribute to the conversation. On November 1st, 2016, the NIEHS celebrated its golden anniversary. This marked 50 years of federally funded research to discover how the environment influences human health. I can tell you we've made a lot of progress in those 50 years. We've gone from studying one chemical at a time to studying all environmental exposures, including chemical mixtures, infections, diet, lifestyle, and studying them across the entire life cycle. In short, we've entered the exposome. At NIEHS, we believe that exposome research is essential, and it's a big focus of our strategic plan. The research we support is broad. We've collected questionnaire information on exposure and also on biospecimens. For children's health studies, we've recently launched the CHEER resource to help investigators measure known suspects and conduct metabolomics research to discover environmental factors which affect our health. In just a moment, you'll hear from Dr. Gwen Collman, who oversees the NIEHS grants programs. She'll share exciting details about our research priorities, and how we can work together in pursuit of this important, holistic scientific endeavor. I believe this will give us the best chance of preventing disease because, as I like to say, you can't change your genes, but you can change your environment. Thank you, and I hope you have a terrific meeting.
Okay, thank you very much. It was very kind of Dr. Vermund to give us this welcoming. And so we're good in time, although we're going to steal a few minutes from Dr. Collman. So we have the 3 talks and then we have the coffee break. I'm chairing the 3rd session. And it's a great pleasure to introduce Gwen Collman, who is the director of the NIHS Division of Extramural Research and Training, where she leads approximately 80 professional staff in the areas of scientific program administration, peer review, and the management and administration over 1,000 active grants each year. She directs scientific activities across the field of environmental health sciences, including basic sciences, organ-specific toxicology, public health-related programs, and training and career development. She also oversees the implementation of the Superfund Research Program and the Worker Education and Training Program. So, Dr. Collman, the title of her talk is Exposome in Revealing the Environmental's Role in Health.
Take that away. And then how do I do this with the clicker? And is it a pointer too? Okay. Got it. Okay. Can you hear me? Can you hear me? No? Can you hear me now? Is that better? Yes. Okay. Good afternoon, everyone. It's really a pleasure to be here at Yale. I was reminiscing earlier. I think the last time I was here was maybe 15 or more years ago, so it's really a pleasure to be here, to meet the new faculty in the department, to be at the School of Public Health again. And you know, today's symposium is important to me as an environmental epidemiologist. I've been working in the field for so many years, worrying about exposure assessment as a critical component of our ability to study risk across populations. And you know, in my career lifetime, we've gone from simple questionnaires to collecting job matrix kind of things in the worker environment, to sampling and measuring things at the monitoring level, the discovery of biomonitoring and measuring chemicals in biospecimens has been a huge explosion, and this concept of bringing all of that together brings so many opportunities and so many challenges. And I have lots of slides to tell you all that those entail, and so it's my pleasure to sort of kick things off, to give you a conceptual framework, a little bit of history around how the concepts of exposures have developed, and to talk about what we think are the opportunities for the future, but also what the challenges are. And we hope that you'll be inspired by the talks today to be able to make the contributions to keep this very important initiative moving forward. So I'm going to multitask here. Okay, so I have too many things to cover in a short amount of time, and I believe in speeding through everything. So I'm going to talk quickly. There'll be a lot of information on the slides. I'm going to make the slides available, and I understand that the video will also be available, so if you want to go back and look at anything. But I wanted to give you a very comprehensive overview. So I'm going to talk about what we mean about the exposure, some initiatives in the United States and in Europe, I'm going to talk about a workshop that we held now about a year and a half ago, and some of the critical areas that we explored in this workshop: biomonitoring, biological response, implementing the exposome in environmental epidemiology, and the big challenge of big data that comes from all of these activities. And then I'm going to tell you, as Linda mentioned, about a new resource that we've developed at NIEHS for the scientific community to add exposure assessment to their already NIH-funded studies. And so hopefully some of you will want to take advantage of that resource, and I'll tell you how to do that. So what do we mean by environment? Well, you know, we can say everything is the environment, and at the most broad level, we're really talking about everything except for genetic factors. But of course, we're thinking about environment in terms of G by E, so we're going to include that as well. Traditionally, NIEHS has supported things about research in the area of chemicals used in materials and the buildings we inhabit, industrial pollution in the air that we breathe, pesticides, medications, cosmetics, and personal products, chemicals we're exposed to in our workplace and in our communities. But we also consider lifestyle factors such as nutrition, smoking, and pharmaceuticals, and the overall aspects of stress to be part of our physical and social environment. This is one of my favorite figures, and this comes from a paper that was published in EHP a number of years ago. This is 54 pregnant women who had samples in the NHANES study. And if you look at the different colors, they represent different classes of environmental chemicals. And so, you know, we always talk about how we live in a soup of exposures, and we all are exposed to many things, and a lot of our work is looking for the effects of one specific chemical by itself controlling for everything else. But I think this graph really says we are exposed to so many things at a time, and in each of these classes, there are many chemicals within each class. So there's nobody in this group of 54 that has no exposures. And there are some that have few exposures to cotinine because they are not exposed to cigarette smoke specifically. But if you look at the other classes, it's out there and everybody is exposed. So the one by one chemical approach is, you know, somewhat old school, and we need to move towards a comprehensive way for us to look at mixtures and to think about all of the chemicals that we're exposed to in our environment. When are we exposed? Well, across our lifespan. In every single developmental window, we have unique and important exposures. And some might say that the environmental exposures during pregnancy, when the fetus is most vulnerable, might be the most important. We do know that there are health effects in childhood from exposure during pregnancy. We know that there are health effects during adult life. This doesn't discount exposures during these other windows, but it does make us put everything that we think about in the context of windows of susceptibility and windows of exposure. So how do we get exposed, where we're exposed, what are all of the factors that impact our exposure levels are in these slides here. As you can see, it's quite complex. The classes of exposure, the sources of exposure, the places where we are during different parts of our life. We spend more or less time in different locales and different microenvironments. The timing, the contact, how does it get into our body, through the skin, through the lungs, through our food, lots of different ways. And then which are the targets, which biological systems, what pathways, which organ systems are actually affected by these chemical exposures or other exposures. So these two graphics here give you a sense of the complex interplay between exposure source and its impacts. So let's start with thinking about the exposome. So back in 2005, 2006, the idea of this totality of exposure and how we could characterize it became apparent. Chris Weil, who is now the director of IARC, put out a paper, I think in 2005, where he described his vision. So all life course exposure, environmental exposures, from prenatal all the way onward, including all internal body processes, external exposures, and lifestyle factors. Well, that was a lot to think about, right? Because it was not simple, but it was simpler to think about things in their own little compartments. And here he was proposing for the first time to think about some summary measure or summary analytic approach that would tell us how these things changed over time and what would be the cumulative impact of all of our life exposures. Since then, many other research groups have thought deeply about this, and there have been a number of papers published. Slight twists on these things. I won't go through all of these individually, but you're probably familiar with the literature. I'm just summarizing here the different approaches. They're really the same thing. What we want to know is, we want to know everything that has impacted our biology, and then how it's related to the health risks that we have as a population and as we have as individuals. So the common elements of this exposome concept are here. So one common concept is measure as much stuff as you can. Measure it by questionnaire, measure it by sensors, measure it by monitoring, measure it in the body, measure it outside the body, measure it in the environment. And measure all the stressors, not just a few. Identify those stressors which impact health. So correlate in some way those external or internal factors, how they impact the biology, the biological pathways of interest, the systems of interest, the things that we know impact certain diseases that you're interested in. Lots of different ways to do that. We have done it in the past by thinking upfront, using the literature to focus in on things that we are known to impact. But let's step back. Let's take an agnostic approach. Let's do untargeted work. Let's let the data tell us where we might go to find things that we weren't looking for. We might discover some new things. And this also is an approach where we can reuse data and we can reuse samples that were collected for one purpose for something else. So seems like that's a good idea, that we could make the most of what our investments are and learn new things about exposure. And then integrate all of this across multiple scales. Multiple time points, multiple exposures, and multiple responses. So I think this is the distillation of this huge challenge that Chris Wild and others had given us. Okay, so how to measure exposures. Well, we're all probably very familiar in our own work with targeted analysis and maybe untargeted, because I know you have a big metabolomics program here. But we want to think about using all of these in some way in our studies and then bringing all of that data together to understand as much as we can about the exposure scenarios. So targeted refers to more classical or traditional biomonitoring, things like the panel of analytes that the NHANES study at CDC are analyzed for, the different classes of chemicals that I showed you in the first slide that we were able to see everybody's exposure types. And then I go to the bottom here. Let's see if I can do this. I go to the bottom here, the untargeted, so using metabolomics or other omics outcomes without starting from what we know, just running samples, looking at profiles and patterns, and then inferring from what we see what peaks might be of importance, and then trying to understand what those are. Creating methods to validate and to then use new analytes in perhaps a more targeted approach. And then I think this is an in-between approach, semi-targeted, where people have pulled together lots of kinds of information from chemical libraries or the literature, different lists of exposures that are known or suspected, and use a variety of approaches to expand the list of targets that we might start with, develop biomonitoring methods, but it also has a bit of the untargeted or unknown approach there too, so that you're not just ending up with that same list that you would have started if you were only biomonitoring. So our institute has invested over the years in expanding the ways we can measure our environment. As I said, we started with questionnaires. Now we have moved into developing sensors and personal monitoring. We want to go from community-based monitoring, like in the stationary air pollution world, to more personal monitoring, wearing these sensors in a backpack, putting them in wearables on your body, and also having these devices integrate things like breathing rates or heart rate, some biological response, as well as giving us some target information on the analytes. So we have invested a lot in things like PM monitoring for air pollutants. So you can speciate the different particles from the filter, but at the same time you're getting information about the child or the study subject, about their breathing rate, their heart rate, some other aspects, so you can already see the integration of the data as it's coming out of the sensor. So we want to move from what we used to call exposure biology, and exposure biology was a term that NIEHS, I think, invented to talk about moving from traditional epidemiologic exposure assessment into tying exposure to biology, not only looking at the sensors for exposure assessment per se, but to look at the biological response as part of that paradigm. So here are tools for multi-analyte exposure, making the biomonitoring easier, moving it in the field, doing it in more rapid real time, the integration of temporal and spatial variation, because a lot of times we were measuring exposure one time, one point in time, one place, and using that for data that would describe a big, much more comprehensive exposure characterization for an individual subject. We want to integrate the internal and the external exposed responses, and then we want to do research which translates all of this. We want to let people know what their exposures are. We want to let them know what they mean, give them the power to do something about their exposures once they have this information about themselves. So this is just another synthesizing idea where we're looking at the complex exposures measured in lots of those ways that I just talked about. We want to incorporate the mechanistic responses in the biology, so we're measuring those through different measures on the same biological specimen, and then we're factoring in the susceptibilities. So is it genetic susceptibility? Is it geographic susceptibility? Is it a time window susceptibility? All those things are part of this. Oops. And then, over time, we're looking at how these things change and then trying to predict certain health outcomes from this new, robust data set that we're creating. Okay, so now I just want to take a little bit of time to sort of speed date through a bunch of studies that are happening around the world. I'm not going into a lot of detail because some of the speakers that are going to follow me are going to talk about them in much more detail, but I thought I might just tee them up. And the reason we're talking about these is because we as an institute have been collaborating and are part of these efforts globally. We're trying to keep track of what's happening and trying to pull a community, a practice of researchers from all of these different areas so that we can continue to advance the field. So there are a number of European groups that I think are a little bit ahead even of the US effort. There's been funding through the the European Union's Framework 7 program to incentivize really intensive exposure assessment in the helix, and exposomics study. Some of them have brought together existing cohorts or looking at things in an integrated way to be able to assess exposure over time. Some of them have chosen different cohorts that describe and help you go deeper into certain characteristics of the exposure-disease relationship. The HEAL study is based on several large population surveys and have developing this integrated methodology for compiling and organizing this data, very transportable to other work after they report their findings. The HELIX study, Human Early Life Exposome, really gets back to my issues of the gestational exposure period and how important that characterizing that exposome is for understanding later adult or childhood or adult risks. And the last is the Exposomics project, which is leveraging long-term European cohorts and their stored biospecimens, again, integrating external exposure measures as well as the internal exposure measures and helping us create a treasure trove of methodology so other researchers around the world can be doing the same thing in their work. In the U.S., we have the CHEER resource, and Linda mentioned this, and I will tell you a lot more about this. This is a network of laboratories that NIEHS has forward funded in order to allow researchers who have NIH grants to be able to send their specimens to our laboratories and receive free exposure assessment to add to the wealth of data that their studies have already had. So for groups that want to have like another hypothesis related to chemical exposure, this is a way to do this without any financial burden to the investigator at time. We have an NIEHS Center of Excellence called the Hercules Center, And at that center, it's at Emory, and we have a speaker talking more about the research being conducted there. But we use these kinds of centers of excellence to highlight methods development, pilot testing, and community outreach to understand the concept of the exposome and to really broaden the expertise that's out there and really encourage them to be the group of thought leaders in the field to help us move forward. Another sister institute at NICHD, the Child Health Institute, has an intramural program called the Longitudinal Investigations of Fertility and Environment, the LIFE study. And again, this is a preconception study where they're looking at trying to understand how the exposomes of the mothers and the fathers impact fertility and birth outcomes. And it was really designed with a comprehensive exposure analysis plan in order to move towards using methods that help them integrate that data and not just continue to look at it, the exposure outcome one step at a time. And I think that intramural program is really doing a lot of really good work there. Our last example here is part of our Children's Center at University of California, Berkeley, where they're looking at, again, wide-ranging exposure assessments, both questionnaire, dust measurements, ambient measurements, as well as biospecimens, looking at childhood leukemia and trying to pull all of those approaches together under the leadership of Steve Rappaport and Martin Smith, who were one of the early thinkers in the exposome story. So back in January 2015, NIEHS, under the auspices of our strategic plan, we have a cross-divisional focus on exposome. We held this workshop. And we brought together people from a number of different disciplines that we thought were necessary to think creatively about how to define the exposure once and for all, because there was a lot of confusion, and to really look at these 5 domains here and put together the best thoughts for where the strengths, the weaknesses were, where the challenges were, and make some recommendations to help us continue to propel forward. So 5 manuscripts were published. And I encourage you all, maybe as part of classwork here, to go through those. They're actually quite nice reviews of the science at that time with some meaty recommendations in each of the areas that I'll go through. There's so much information in each of those papers, I can't really go in depth on any of them. So maybe that could be a scholarly follow-up from this symposium. So let me start with biomonitoring. So again, we talked about the traditional biomonitoring that I think all of you are quite familiar with. How do we go from those traditional methods of targeted analysis to a broader, more comprehensive, including the known and the agnostic approaches? The untargeted analysis, some people just think about metabolomics as measuring the exposome. That suits some people's definition and how they frame the questions at hand, but I would say that it behooves us to do all this type of analysis on any given specimen, take data that we have from questionnaires in studies, do other external monitoring, and try to bring them all together. It's a big ask. But so the working group at the conference decided to talk about the advantages and disadvantages of the traditional biomonitoring approach, and I've highlighted here. I've highlighted here, some of the key aspects of the advantages. So, we have reliable methods. They're tried and true. We have a lot of data, like from the CDC and other laboratories, and it's comfortable, although expensive, to be able to do biomonitoring. And it's been a little bit, I would say, democratized too, because there have been some state health departments that have taken on biomonitoring programs so that they are able to know the range and profile of environmental exposures in the communities. California is an example of a biomonitoring program focused there. So then that means that there's some reference data from your state or from the NHANES data to compare your study data to. But there are some disadvantages. You're only looking at that select group of known chemicals. It doesn't go beyond that. And once you test that hypothesis on that one chemical, maybe you're done, maybe you're not, but it's not a comprehensive look at all exposures. And as I said, it's expensive and time-consuming. In general, it requires pretty high sample volumes, although over time, we have been trying to reduce the size and the quantity of the volume of the samples. And there has been some progress there, but maybe not enough. So let's now think about the exposome approaches and why are they valuable? So they, I think, are very valuable for emerging exposures and emerging toxicants. It might be, if you have an environmental disaster of some kind and you don't exactly know what blew up at a plant or where there was exposure, you might try an agnostic approach after you collect biospecimens because you're trying to discover what the exposures were. This approach would help to identify the emerging concerns. We want this to link the exposure to the biology, which is a step beyond what the targeted analysis can do. With new methods being developed, it may require smaller sample volume to sort of shoot through your machine and get a lot of data. We get a lot of data. We don't always know exactly what all that data mean, but that's part of the discovery process to identify which of those data points are important and then what we do with them. Some say that this may not be an optimal approach when some of our exposures might be at extremely low levels. We may miss them when we use our sensitive machinery, but that's yet to be determined for the full class of chemicals. These baseline values may not be able to yet be defined because we don't have reference standards for things that we don't yet know we can measure. So there's that whole process of doing that. And it relies heavily on library searching of spectra for annotation, standards for confirmation, validation, and then developing methodologies to go back and add those to your list of targeted analytes so that you can measure them in studies in the future. Okay, so I want to give you just a few examples of this in the research world in practice. So this comes from the Emory group as well, part of the Hercules Center, where they were doing a study of arsenic effects, and they in a Chinese cohort, and they identified 5 biomarkers that they hadn't really been looking for, and they were in biological pathways that were emerging in the literature as possibly important. So, they were able to measure all of the arsenic-related measurements, and they probably could have done most of those through targeted analysis, but by using a metabolomics approach, they were able to be able to identify testosterone, guanine, hippurate, and another one that has a really long name that I can't pronounce, and serine from 61 candidate metabolites. So again, it gives you a lot more information, and then you can go back and dissect the relationships between those chemicals and the biological pathways. This is a brand new study that I actually just learned about 2 weeks ago. In California, they're studying women firefighters who have known exposures to a host of chemicals through their firefighting work. And there is a perceived excess of breast cancer in the firefighter women in San Francisco. And so through partnership between a breast cancer advocacy group called Breast Cancer Prevention Partners, UC Berkeley, and the women firefighters, they have started an exposure study to be able to understand what these women were exposed to. So they're starting with some ideas, traditional things like flame retardants and PAHs, but they're also interested in other chemicals that might be in the environment where they're fighting the fire, and also might be part of the tools and instruments that they use for fighting the fires. The foam that they use, the volatiles that come off of the fire. So they're using this hybrid approach by bringing in information from chemical libraries, looking at things in household chemicals, because remember, these women are using products in their day-to-day life, so they have those exposures. They have the exposures that they have on their job. They may be exposed to pesticides or endocrine disruptors through their diet. And so they're thinking about how to integrate all of those streams of data when and if they see that in those biological specimens that they're taking. So I thought it was a very interesting process that they went through to identify a bunch of chemicals, deciding which ones they would eliminate based on technical ability to measure them, and then to go forward with a schema to go from this really big volume of potentials to narrowing it down for chemicals that they wanted to do for validation. So I can't step through every single part of the process, but you can see they're going to go from a large number of possible unknowns to they right now have the resources to do about 10 final, best targets for validation, and then they might go back later and do some more extensive biomonitoring with these new targets. Anyway, I think it's a really interesting application of an exposomics approach to understanding. And here's one more study that really focuses on the pregnancy exposome. We've talked about this, in the context of some of the other studies that are already in the field, studying pregnant women following their children. And this gives you, using a very creative way to show you the associations, the correlations between the various chemicals. But these chemicals are only part of the story, right? So they have things in here that are byproducts of some of the exposures here, but things like noise or social factors or sociopolitical factors may be also enormously important in the environment of a pregnant woman that may relate to a health outcome in their child or in themselves, over time from those early life exposures. So, I think they're at the beginning stages of understanding how to integrate all of these pieces. This is work I think that Dean has done, and I'm only putting it up here because I'm not going to go into the details. His talk will tell us a lot about this. But the point of this is to say that you can measure, he's developing methods where you can measure a lot of chemicals, a lot of peaks, a lot of metabolites in a small volume sample. And that is a huge improvement over what we're used to doing, where we were really focusing using precious biological or urinary samples to measure one thing. I mean, it's like day and night, right? Such an amazing advance. So I look forward to hearing more details about that. And then this is a complicated slide as well, but this is an example of endogenous exposures. So, the microbiome is a really important part of our physiology and an important part of disease risk. And we're learning more and more about it every day. And those bacteria, they generate all kinds of products, and they interact with environmental chemicals, and they interact with physiologically present other factors in our system. And using some of these metabolomic processes, you can think about the microbiome exposome, and how all of those factors may play into helping us understand these complex interactions. So it's an interesting paper, and it would take a lot of time for me to go through step by step, so I encourage you to look at it. So, in summary for the biomonitoring challenges, we have an abundance of analytes. Some of them are not that present. We are looking for small things, new things. We have some challenges related to lab capacity. Not every laboratory in this country has that ability, but we do have some regional metabolomic centers. We have our NIEHS core centers that have a lot of metabolomics capacity, and for NIEHS, we have our CHEER network where metabolomics can also be part of this, as well as the targeted analysis. We want to really strive for accurate and reproducible measures, no matter what we're measuring, so we have to make sure that our research gets us there. We want to we want to take that data and put it out there, so we create databases, but we also want to use already existing databases to help inform us for the things that we're looking for, or to help us understand the relationships that we may see from the analysis. And we need new bioinformatics tools to be able to make that happen. These are the group recommendations for biomonitoring, and I'm not going to go through them all because it's a little bit repetitive of what I've been talking about. But again, there's a paper published in EHP that will detail those. The next area I want to talk about is the biological response part. So we go from thinking about the chemicals to the biological pathways and some of the technology that we already have available to us. And I think the key message here is that we want to measure those at the same time we're measuring our chemical exposures. And then we want to integrate that data when we're thinking about how exposure affects our health. So here are some pathways that I'm sure are very familiar to all of you. These are omics galore, right? Every omic you can think of, and some that we haven't thought of yet, can be very informative and important as we're looking at the exposure-response relationship in many of our study populations. So just a quick example, this comes from UNC, Rebecca Fry's lab. She's conducting a study in Mexico called the Biomarkers of Exposure to Arsenic Cohort, or the BEAR study. And she's particularly interested in proteomic shifts as a result of arsenic exposure, and she has samples from cord blood from 50 newborns in this study. And you can see, the reason I put this up here is because she's using a familiar-looking heat map to try to show us where the peaks, where the valleys are, where their exposures, helps inform us to go deeper and to try to tease out some of those relationships. Okay, some of the really pressing questions when we're thinking about the biological responses, you know, which disease outcomes of interest are we looking for? Which of these pathways are known to be associated with disease pathogenesis? How do we hone in on those outcomes of a concern, and whether there's an influence of the environment on those? So there's a lot of things to think about when you're planning which of these things to include in your study. Here are some recommendations from the Biological Response Working Group. They really have encouraged a systems and pathway-based perspective to demand reasonable and biological feasible explanations of what we're seeing here, not just really like create a new litany of associations that maybe, you know, are false positives or don't have a lot of anchoring in the biology of the disease that you're looking at. Think about these sample collection issues. Make sure you're really able to do these omics techniques on the specimens that you've collected. Okay, so environmental epidemiology, as I mentioned, is the field that I grew up in, and so I think about this quite a bit in the context of all of the work that we do. And the exposome is a really tough concept for us epidemiologists to wrap our heads around, because we really are comfortable in the one chemical, one exposure thing, and we want to control for all those other complicating factors. Well, we got to stop that. We got to move away from control. We got to get into the how do we understand the interplay between them, and how do we use that, you know, in our traditional models where we're looking for odds ratios or risk ratios in our epidemiologic studies. It's a hard transition. We're all struggling, but I think we can get there with the help of our other colleagues who can help us understand the methods and really to create the right statistical tools so that we're able to really analyze this data in a new way. I think the most important part of the contribution of this group was to remind the exposomers about confounding and to make sure that we assess for confounding when appropriate, and that we don't, but we do need new models of thinking about what confounders mean in this complex interaction of exposure to disease. So yeah, we really want to, you know, incorporate this, and we want to move on. So the, you know, again, just a few studies. So the Japanese Environment and Children Study, it's a national children's study in Japan. They've moved very quickly to enroll 100,000 expectant women, and they're going to follow these kids for a super long time. And at the get-go, they're measuring exposure at multiple scales and multiple levels, and they have a commitment to work on the analysis of this in that get away from the one exposure, one effect model, which is a very exciting thing. They will be able to, you know, characterize these early life exposures with children's health outcomes, and you know, it's really a model study for the rest of the world in terms of developing these cohorts. The life study I've already talked about, so I'm not going to go there. So again, integration, merging data across the different cohorts, focusing on the windows of exposure is a critical piece for epidemiologic work. So not to think about your birth cohort study or your epidemiologic longitudinal study by itself. It's always had value for the group of investigators who have been studying. But you have to think about it now in terms of the bigger value that it has to the cluster or the group of cohort studies that might be pulled together in a meta-analysis or an integrated analysis to be able to look at this. So it's very important to use standardized or comparable exposure measurements and phenotype outcomes and then link the omics work that you're doing across the cohorts so that they're done in a similar way, so we can pool all of this at some point in time. That brings me to the Phoenix Toolkit. I hope you all know what this is. If you don't, it's a very large effort that the National Genome Institute has started. It's available to anybody, and I would encourage you to go to this website. What it does is provides a standardized way of measuring phenotypes and variables for other factors. So there is, within Phoenix, a section on environmental exposures. Now, every investigator I have ever known in my whole life wants to measure that thing their way. And they want to use questions in their questionnaire, their questions, because they're comfortable with those or they have some experience. This gives you guidance for a more standardized way to do that. So maybe your questions will look like the questions of the other studies and will give us a better opportunity to combine them all. There's every disease endpoint available here and many, many exposures, not just environmental exposures. I highly recommend it. It's really a great project. And there was a recent paper in 2014 which used Phoenix measurements in an EWAS type study for type 2 diabetes to look across all of these different exposure variables looking for the risk of type 2 diabetes. So it's not so much the importance of what they found, whether it was a risk factor or not, but I bring it to your attention because of the strategy. If we're going to use these standardized methods, we have to make sure they mean something, and we want to use them in studies where we can make comparisons. So I think this is a good addition to the literature there. So this is a classic study that our colleague Chirag Patel started back in 2010. I can't believe it's that long ago already. This really changed how people thought about environmental exposure assessment and how we use different visuals for combining and integrating our thoughts around this. You know, he took Manhattan plots from the genetics world and took our exposure data and flipped it around, and you know, many of us were like, whoa, that's amazing. And I think it brought a lot of innovation into the opportunity to think about using your data in lots of different ways to show how show what the associations look like and look for different outcomes in there. So I think it was a great beginning, and I'm sure he'll talk about this and other related things in his presentation as well. So challenges to incorporate the exposome into epidemiologic research. Well, you know, people say there's a lack of infrastructure and coordinated support to be able to do this. So I think, you know, my institute, the EU, others are mindful of this, and we're trying to think about how to provide that kind of infrastructure to enable this kind of work. It's still very complicated to characterize the biological and the temporal variability that we have in our data, in our studies, and especially in our longitudinal pursuits. And there still is a lack of data, tools, and methods. We're just going to have start at the end of this fiscal year a new consortium related to mixtures. It's called the PRIME program, and the purpose of that is to develop and expand statistical tools to be able to be looking at more than one chemical at a time in these types of studies. So the last area is data. And of course, as I said, there's so much data that comes out of these analyses. I mean, just one run on the mass spec, man, there is a ton of data. And large epidemiologic studies, we are used to lots of data, but we don't use it all in a way that really brings the investment. So we need ways to generate those data. We need to store it, manage it, analyze it, and we need to share it. We need to understand that no one single research group can possibly have the capacity to really utilize all the data that they generate, and that even if they do, they're seeing it from one perspective. And so when you open up that data to a larger field of community of researchers, you can see that data from lots of different directions, and you can bring much more out of those data than what we had first anticipated. The comparative toxicogenomics database that Caroline Mattingly at NC State develops and maintains for the community is also an important resource for us to use when we think about these exposomic studies. It's a curated database which pulls together literature that helps to show these relationships. And then last year we added some funds to her work to have an exposure piece to this curation. And so you can go in, there's an online tool, you can try to pull together a curated literature around the topics that you're interested in studying, and it can help you understand what you're finding in your data, but could also help you generate new hypotheses to study. Excuse me. Our CHEER work, we do have a requirement that the CHEER data that's developed will be put in a database that will be shared. And so we're creating an ontology and a shared vocabulary. We want our exposure variables to mean the same thing that others. And so NIEHS is very mindful, and we want to take a leadership role in creating this vocabulary with the field so that others will use it, and that so your data, when it's reposited one place and it has toxicology data or environmental exposure data, those data variables mean the same thing across all of the studies. So everybody understands that there are a huge number of challenges to these big data efforts, and I'm just acknowledging that they exist, and I hope that some of you here in the audience will be inspired to work on them. So there are some recommendations from this group as well, really with a great focus on how to share the best data and reuse it and recycle it for the best one. So in my last few minutes, and please tell me when I'm done because I could go on for a while here, I want to just quickly give you some more details about the CHEER program, because I'm so excited about this. 10 years ago, we thought about what infrastructure would be necessary to really galvanize exposure efforts across our field, and it was proposed that there had been genome centers when the Human Genome Program had identified the sequence that investigators could go to with their specimens, and they would sequence, because sequencing was really expensive and not everybody could afford that. Well, exposure biomonitoring is expensive. It's a little less expensive now than it was, 20 years ago. But not everybody has the capacity to do that. And the line to line up at the CDC is really, really long. So not everybody even has access to our national biomonitoring places. And so how could we as an institute provide that to the community? So we're specifically interested in this pool of scientists who are studying children's outcomes, but have never considered the role of environmental chemicals in their studies. We didn't need to preach to the choir of our children's environmental health researchers. They get it. But the other investigators, if they're studying asthma or diabetes or cardiovascular disease, they're mostly looking at risk factors about lifestyle, about demographics. More traditional epidemiologic variables. And people are a little bit afraid. They're intimidated by this concept that environmental chemicals could be in their causal pathways. So we wanted to eliminate those barriers and provide that opportunity. So I hope some people that are in the room today and maybe on the video cast are not environmental health scientists, but want to learn that there are resources now for them to consider these hypotheses. So we created the CHEER resource. We have a coordinating center to help us move through the process. We have a laboratory network, 6 laboratories across the country that are able to do all of this targeted and untargeted analysis, biological response, environmental chemicals. I'll show you the list. And then we have a data repository, which will provide a place to store the data that's generated by CHEER in a standardized way with shared vocabulary. We'll also provide statistical consultation. Can do analysis for investigators who don't have the capacity or can just support their analysis. The data can go back to their home labs, and you can do your own analysis, or you can do it through the CHEER portal. So we've made it pretty multi-purpose depending on what people want to do with it. So here's a list of the investigators who are in our network. Here's a list of all of the targeted analysis that we can do. And you can see it's not just chemicals, it's hormone panels, dietary factors. There's a lot of tobacco-related biomarker work at Minnesota that's part of this. So it's bigger than just sort of endocrine disruptors or things that are really associated mostly with our institute. There's a couple of ways to do the untargeted analysis in our network. There's the biological response indicators, and then each of the labs has the opportunity for new methods development as well. As we go through the 5 years of funding, there may be some new analytic things that are worth working on so that we can process more samples in a timely fashion to keep the workflow going. So again, as I said, you can get targeted work, you can get untargeted work, you can get biological response variables. We are the exposure center for the new ECHO program. You, I think, are familiar with that. It's the environmental contributors of determinants of health in children. This is a group of 30 cohorts. Yeah, 30 cohorts. Oh, no, 84 cohorts. There are 30 study sites with 84 cohorts around the country. All of these cohorts had to bring to the table biological specimens that they had collected during pregnancy or early childhood. And our cheer labs will be able to analyze an agreed upon set of analytes and do untargeted analysis for all of the specimens so that we can have one database for the whole program, which will have all of this work. Each of these cohorts, have their own questionnaire data, and they may have other sampling data depending on what the purpose was. And we hope that we'll be able to pull all of that together for the best analyses for the questions at hand. This is just the data repository. The data center is a really important part of CHEER. It helps us think about the big data challenges of the exposome, and it's our attempt to sort of create prototypes and processes that we will share with the greater environmental health community for them to be applying to their exposomics work. So how do you apply? So you're eligible if you have an ongoing or completed epidemiologic or clinical study on children's health. In any area, any endpoint, if you want to add environmental exposure data to your study, or you need more extensive analysis of exposures to support your hypotheses. If your study was funded at least in part by NIH extramural funds any time in its lifetime. So you do not need to have an active NIH grant right now. You just have had to have support by the NIH. You're eligible to apply. Some institutions are not eligible to apply for NIH grants. And your study has existing samples that were collected from children or their parents, and or if your studies have not yet collected, you can apply to CHEER for consultative services on how to collect those over a next grant period, and then get the analytic services from the CHEER Center at a later time. You have to agree to share the design details and the supporting data, and you have to agree that the data that's generated by the CHEER labs goes into the CHEER data center. This is the website for applying for CHEER. I also have copies of a one-pager that I'm happy to leave with you today, and you can refer to that. It has contact information for our people at our coordinating center. We review these applications a couple of times a year and make decisions about which of them are going to go forward for these resources. And let me remind you, you do not have to pay for these. Okay, this is all pre-funded. We made the awards to the labs. It's all in the laboratories funding pool. People who come forward for any of these services, it's free. And free is good. So just to wrap up, I've covered a lot of cross-cutting themes related to the creation of the exposome and its implementation. I tried to give you a framework for what's going on internationally, although I appreciate that it was very quick and very superficial. We really want to train a new generation of environmental scientists to start working with this concept and incorporating it in their work from the get-go. We're good with changing people's ideas and moving them into a new area, but the training of people in all of these components, the targeted analysis, the untargeted, the questionnaire standardization, the harmonization of data, and the big data and the tools are essential for the future of the field of environmental health sciences, and essential for us really realizing the commitment that we all have to this exposomics approach. So with that, thank you for inviting me, and I'm happy to have been here.
Thank you very much, Gwen. Unfortunately, we're running a little bit behind. We can have the questions during the coffee break for Gwen, so we can give the floor to Dean. But before, I would really like to bring to your attention that this is what this department is going. We're going to the 21st toxicology and environmental epidemiology to address all the health issues. And I really like your slide talking about not using the metabolome, not replacing the environmental epidemiology, but complementing. And this is one of the reasons that I came to Yale to really develop this two disciplinaries together. And by the way, we're going to have Toshiro Kawamoto next year visiting Yale. And Toshi is the director of the Japan Children's Center, and he's a very good friend, collaborator of mine. With that being said, it's a great pleasure and a big honor, actually, to introduce Dean Jones, the world expert on redox proteomics, detailed metabolomics, high-end metabolomics, whatever word you can put to metabolomics fits to Dean. And Dean is going to talk to us about daily exposures, lifetime memory, sequencing the exposome. Dean?
All right, great pleasure to be here. Always a difficult act to follow to be after Gwen, but I will do my best to keep you awake during this period of time. You okay on this?
Yes.
All right. All right, good. All right. So this is not a cummerbund. I'm not wearing a faked tuxedo. I just had back surgery and I'm still recovering, and it's just in case I fall off the stage. Gwen has already introduced this, that I'm a member of the Hercules Exposome Research Center that's directed by Gary Miller, and we've been working together for many years on this. I will note that we have had really very important funding from NIEHS over the years, but this has been a very expensive effort, constantly fundraising, and we've had a lot of support from the different institutes in National Institutes of Health, as well as Department of Defense, EPA, and other sources. When you get enough gray hair, or you lose all of your hair, you have the opportunity to talk about history. And I put this slide in here really for those of you who are not of the young trainable generation, but the retrainable generation, or generations, because really we have to continue to learn throughout our careers. And about, if we go back in history, I'm trained as a biochemist. Always have been a biochemist and always will be. But I was trained in molecular toxicology and nutrition. And my career in the 1990s was really on mechanisms of cell death, studying apoptosis and really oxidative stress. And I put this here because oxidative stress research fell off a cliff at about the year 2000. And that was because there was this minor problem. There'd been $1 billion invested in double-blind interventional trials with free radical scavenging antioxidants, and they showed basically no health benefit. Now, this is disaster if you are a biochemist studying oxidative mechanisms of cell death, and all of your experimental studies are showing you that this is the most important thing, and now you have basically everything just falling flat. And so that was where I was in early 2000s, was really that issue. And we had been studying at the time thiol disulfide systems. I'm not going to give any detail on this, but the key point was that this concept of oxidative stress being a simple balance, the free radical scavenging trials showed that it wasn't really of that much importance in terms of human health outcomes. And our studies were showing that things are never in balance, that in fact there's this redox interface between what we call the gene-environment interface that really lies here between the genome and environment. And so we probably need to think about oxidative stress in these terms. But really more importantly, if you step back from scientific method, scientific method is, we all think about the hypothesis-driven research. But the usefulness of hypothesis-driven research actually goes back to the first step in scientific method. And the first step in scientific method is observation. And so the problem is that if we don't have good observational tools, then we're very limited in what we can hypothesize. And so when I was confronted with this problem in 2002 and early 2003, what was I going to do with the rest of my scientific career? Was I going to return those NIH grants to NIH? Or was I going to rethink how I was doing things? And so what I decided was that we really needed to rethink what we were doing. So at that time, I was part of an Emory team that was sent up to NIH when when Zerhouni was laying out the NIH roadmap. And so I went to one of those initial talks by Zerhouni, who was the NIH director at that time. And he talked about data of the future. This was absolutely wonderful. It was based upon the progress in gene sequencing and genomics. But what he said is that data of the future needs to be simple, needs to be high throughput, automated, needs to have extensive coverage, needs to be reproducible, needs to be standardizable for cumulative libraries. These are things that Gwen already just talked about. Needs to be affordable, needs to be cost effective. So as I went back to Atlanta, flying back on the airlines, I started thinking about this. I was trained in metabolism, and so metabolomics was obviously something that I would think about. And recalled that in 1957, there was this revolutionary breakthrough. Moore and Stein developed automated amino acid analysis. This ultimately they ultimately received a Nobel Prize for the application of this. But the remarkable thing was they had gone from 1 chemical at a time measurement to 20 chemicals at a time. Now, 15 years later, I was a biochemistry student, and one of my professors, Mike Litt, very bright scientist, leading biochemical geneticist at that time, was lecturing in one of the courses, and he said, a few proteins have been sequenced, but of course it will never be possible to sequence the human genome. 30 years later, wow. So what was not even imaginable by a very well-trained, educated, sharp, leading scientist was accomplished in 30 years. And so where were we in 2003? Basically a half of a century past the Moore and Stein development. The best analytical labs in the world could measure about 200 chemicals. So only a 10-fold improvement in half a century. Wow, quite a comparison. So basically what I did was I decided that, wow, we need to do better than this. And so I thought, well, let's use Zerhouni's data of the future as a guide to try to develop new approaches to metabolomics. And so in trying to search through how could we do this, how could we transform things, I ran across work by Alan Marshall, who was a co-inventor of the Fourier transform ion cyclotron resonance mass spectrometer, the ultra-high resolution mass spectrometry. And he was active at Florida State University, and so I called him up and said, hey, can I come down and visit you and talk to you about this? And so in one sentence, just one sentence during my visit with Alan Marshall, he convinced me that this was definitely something we should pay attention to. Because what he said was, with FTMS, we can measure more than 20,000 chemicals in crude oil in a single analysis. So, simple extrapolation. If you can do that in crude oil, can you do it in human blood? And so that was what we set out to do. And of course, as all good NIH-supported investigators do, submitted an NIH proposal for this. Wonderful reviews by reviewer 1, reviewer 2, clearly fundable scores. But reviewer 3 had only 2 sentences. And one of the sentences was, we already know how to do metabolomics. We have wonderful triple quad and QTOF instruments that can do that. The second sentence was, this would be a waste of a good instrument. So, unfortunately, the dean of the medical school didn't see this review, or at least he hadn't read it. And so they provided funds for us to get one of these instruments, and that really established our metabolomics program. And so the real issue on this is that 90% of the top 2,000 metabolites in the human metabolome have unique elemental compositions. And so this instrumentation allows you to measure things with such mass accuracy that you basically, for the low molecular weight chemicals, you can pretty much get the elemental composition. So the main point is that if you have high abundance chemicals, you can, in principle, you can measure them based upon the accurate mass, and you only need MS/MS for those where you don't have, that you can't resolve them by these methods. And so there was another point that I wanted to raise about this. So we use mostly the Orbitrap now, but we also have a couple of FT-ICR instruments still in operation. They're remarkable in terms of the accurate mass measurement. But the important thing on this is that it really comes from one of Alan Marshall's articles about the original FT. And that is that the method of detection of ions is fundamentally different from the way ions are detected in other mass spectrometers. And that is, in this instrument, and this is, again, the Orbitrap instrument, and I'm not supported at all by Orbitrap, but I really think that it's an important technology aspect that needs to be paid attention to, is what the Orbitrap is doing is it's orbiting the ions around this central electrode. But the important aspect is, so in terms of the mass resolution, the reason that you get better mass resolution is that in a time of flight instrument, the ions are only moving from basically here to the wall. It's in a reflectance mode, but they're only moving that distance, and so you can only separate based upon the time of flight. In the Orbitrap and in the ICR instruments, the ions are orbiting, and they're basically going the distance of a soccer field. And so what that allows is you get better separation, and that's why you get the better mass resolution. But it turns out that's not the key issue. The key issue is that the way the particles are measured, the way the ions are measured, is that there is in the electromagnetic field, there's an oscillation of the clouds of ions back and forth. And then they're detected as an image, what's called an image current. And so it's the movement of this ion cloud. And in the original Alan Marshall description, what he claimed, and I'm not a physicist, so I don't know whether the truth to this, but what people I've talked to said that this is true, that you can get orders of magnitude improvements in sensitivity by this type of detection. And this is the key point in terms of what we've in our applications. So this is a degradation product of DDT, but it's degradation product of many chlorinated hydrocarbons that we are exposed to, and everybody here in the audience has this in their blood. So basically what you see is it's a very small peak here, but you can't really appreciate how small that is. That is basically, this is 10 to the minus fifth the full scale, and this is less than 1% of the signal. So this is basically 8 orders of magnitude lower abundance signal than the highest abundance signal that you're measuring here. So it's 8 orders of magnitude. It's quite remarkable. But what's perhaps more important is that an analytical chemist looking at this would typically just throw it out as garbage. And the reason they would throw it out as garbage is shown in this example. These are triplicates. These are just technical replicates of the same sample. And look at this peak right here. This one right here on the left, basically the same magnitude as this one that is an environmental chemical. If you look in the previous, not there at all. If you look in the following, not there at all. So basically you have phantom peaks that are occurring that you cannot distinguish those at all from a real signal if you just look at that sample one time. So what we realized is that we needed to run things at least in triplicate in order to be able to have some certainty of what we were measuring, that it's reliable. And so that's really a key aspect of being able to get these low abundance chemicals. I'm not going to go into the details on that, but I think you can see from that it's very important that we think about how the samples are being analyzed. So what we did in terms of following this, this kind of plan, following what Zerhouni's Data of the Future, we introduced a very simple one-step protein removal with addition of internal standards. We initially started with 10-minute analyses. With the improvement in the instruments, we're now at 5-minute analyses. And the first thing we wanted to do was question, is it quantitative? Because if it's not quantitative, we just have to walk away from it. And so we worked with Fred Strobel, the director of our mass spectrometry center, and we tested it in terms of one of my favorite areas was aminothiols, glutathione, cysteine metabolism, and so forth. You see that here. What's important is this is plasma glutathione disulfide, is 4 orders of magnitude lower abundance than the cysteine. And so what we did in this study was we really established that the instrumentation is quantitative, it's reliable, and we compared it to multiple other methods to actually establish that. So in terms of these steps, then we're halfway there in terms of the methods, but we really haven't dealt with coverage, the extensive coverage. And so when we were working on this in about 2008, 2009, Chen-Wei Yu was a new assistant professor in the Department of Biostatistics and Bioinformatics, and we met and just were chatting, and he said, oh, I can write the software to do that. So he's basically, within 6 months, he had written software to be able to enhance the data extraction. He calls this adaptive processing, and it's really quite powerful. And we applied this in an initial study that Jennifer Johnson did as a graduate student in this paper that was in Analyst, a practical approach for metabolic patterns. And I want to emphasize here that this was really patterned upon what Jeremy Nicholson had developed and Elaine Holmes had developed for NMR spectroscopy. It was the same concept. You don't really need to know what the individual metabolites are. You just, if you have good methods for pattern recognition, data reduction, you can get a lot of information. And so we did a series of studies with individuals where the samples had been studied, people had been studied over a 10-day period, 5 days on a complete nutritionally complete diet, 5 days on a nutritionally deficient diet, and we had a total of 34 different samples for each individual. And the question was, does an individual have a unique metabolic phenotype? And the answer really showed that very clearly, with the exception of this is just 6 of the individuals. We had 13 individuals in the study. Only this individual was the only one that showed really a remarkable response to the nutritional deficiency. But otherwise, you could see from the topological map of the metabolites that the individuals really are different, and the method is very powerful. You've already seen this picture that Gwen showed. This is from a review from several years ago now, but basically a single analysis, you get metabolites from almost every human metabolic pathway. It's pretty easy to map those out now. And it was about this time that I met Steve Rappaport and got introduced to the concept of the exposome. And it has really driven our research since. And the concept that Gwen didn't really emphasize, but is very important, is that if you go to Steve Rappaport's papers and others, that the attributable disease risk is really only about 5 to 15% coming from genetics alone, that the bulk of it is really coming from the environmental exposures or the gene by environment interaction. What's important in terms of the exposome is that we really are not measuring very many exposures in terms of our routine analyses, whereas with the genome we have the ability to get tremendous detail in terms of the measurement. And so the need then is to really improve the platforms to be able to measure the chemical exposures. Now the estimates are that humans are probably exposed to 1 million different chemical substances during the lifetime, and so it's quite an incredible array that we have to deal with. So we started thinking about this concept of universal exposure surveillance. What would we need to be able to do in order to really cover the exposome? And if you have high dose exposures and chronic exposures, that's relatively easy to measure. If you have low dose chronic exposure, in principle it's easy to measure, except you have to have the sensitivity. And this gets into that question that Gwen talked about. It's the cost. It's very expensive to do a lot of these targeted analyses. So that becomes a limitation. If you have intermittent exposure, it's much more difficult, because now if you don't happen to measure an individual, well, while they have that chemical still within them, you don't measure it, you miss it. And we've been working with the Department of Defense and deployed troops. If they've had a transient exposure and have now been shipped home, you don't have any chance of measuring what that actual exposure was at all. And so in this case, you really have to rely upon that biological response. And so this gives us a perspective that we have to have really high sensitivity, we have to have repetitive measures, and we have to incorporate this concept of the response to exposure. So Gwen talked about this a little bit. This is from a 2012 review on nutritional metabolomics, but it emphasizes the point that if we're talking about metabolomics, we're really only talking about 2,000 or so metabolites that are products of the gene-encoded proteins and enzymes. But if we look at the diet, the food metabolome is probably 200,000 chemicals. So we're exposed to all of those. Of course, everything we eat is also acted upon by our intestinal microbiome. So we have all of that 200,000 and the products of the microbiome to which we're exposed. But then you put on top of that the 1,000 or so drugs, but then the dietary supplements that many take, again, a very large spectrum of chemicals. And that's even before you get to the nearly 100,000 chemicals registered with EPA for commercial use. It includes all of the skin creams and everything else that we put on our bodies, as well as all of the fabric softeners and everything else that we're exposed to, as well as then the broader environmental chemicals. Now, the point that I like to make on this is that we have 10,000 chemicals in high volume use commercially. Those are a lot of those have been tested, not all of them. You don't have a lot of safety testing on a lot of these because there's no expectation that humans are going to be exposed to them. But each of those high volume use chemicals has contaminants within it. So if you take something, I can't remember now how many millions of metric tons of some of these things are out there that produced commercially. If there's 1% contaminant of that, of one of those, that's completely uncharacterized, that's still coming out into the environment. And so we have a massive amount of unknown that we have to think about. So the main approach that we've been using is outlined here, and it was in a couple papers that Doug Walker has written over the last couple of years. The main concept is that we can use this high-resolution metabolomics platform as a central way to link exposure, internal burden, and biologic response. And basically the concept is that we have exposure assessment, and if we can link that to an internal body burden related to that exposure, and then use this to link it to the metabolic response, then we basically have a single way to link everything together. And so that's been this concept, and this is the main take-home, is that I believe that this high-resolution metabolomics provides a way to do this. It provides a way to unify all of the other aspects of exposome research and give us sort of a central anchor for things. So this is extensively developed now, and I don't have time to go through details, but we have about a dozen different methods, papers, basically going from the mass spectral data through what we call deep data extraction, and then a variety of biostatistics, bioinformatic tools, and then identifying pathways and networks of interest. And then in our workflow, the metabolic forensics is really the last step. So we sort of have an inverted workflow. This is sort of the take-home of where we are. Over the past decade, the analytic capabilities have increased phenomenally. Now we've done feasibility studies for being able to get between 200,000 and 800,000 chemical features out of a single analysis. So it's really well beyond anything imaginable just a few years ago. Where we are in terms of our operations is we set our parameters to about 20,000 is what we work with. So we don't extract this data. It's in the raw data, but we don't extract it. We don't even try to use it, mostly because we don't know what to do with it. It's not characterized. There's no identities associated with it, and the computational time is just too much. It's too expensive for us to deal with it. But in any case, that information is there, and it's capable. We can do that now. So we've outlined a lot of these details in a paper, Computational Metabolomics. Karan Uppal's first author on that. And to reiterate what Gwen said, we have here in our abstract, to this end, there appears to be no choice but to abandon the limitations of measuring one chemical at a time. And that really is the point. We don't have any choice. If we're going to deal with 1 million exposures in a human lifetime, we cannot afford it. It's not doable to do it one chemical at a time. So jumping back to the Zerhouni's description of the data of the future, one of the issues had to be cumulative data structures. How do you do it so it's an open source, basically? And what we did was develop a software, or develop a routine called reference standardization, which provides us the capability of quantification in this very complex data analysis system. And in this case, you can see quantification of glucose in millimolar. You can see bilirubin here at micromolar concentrations. Same analyses. This is non-smokers. You can see cotinine measurements down here in the nanomolar region, and you can see insecticide Perimicarb present in the subnanomolar concentrations. So it really emphasizes that it is quantifiable, and what's important is it's quantifiable over 7 orders of magnitude in terms of what that data shows. So this is one of Steve Rappaport's figures where talking about environmental chemicals exposures are much, much lower concentrations than what we have for drugs and food products. And so this is the enabling capability, is having reference standardization, having something that's standardizable for cumulative library, quantitative over a very broad range, and giving us that extensive coverage. So the last of the Zerhouni data of the future has to do with this affordability. We sort of used as our target $100 per analysis, and that sort of drives that, why we draw that dotted line of 20,000, because we can't, it's not affordable at $100 per sample to be able to do better than that at current capabilities. But what this gives us about 1 cent per chemical for the analysis, and that is doable for a lot of our studies. Now during past year we've added capability, and Gwen also showed this. This has to do with the gas chromatography is better for measurement of volatile and semi-volatile chemicals. A lot of the environmental chemicals are better measured in this way. And so we've been working on this high throughput environmental biomonitoring. This is work that Doug Walker has really been pioneering. And what his data show is that in a single analysis, you can measure hundreds of environmental chemicals using this, and it has the ability to actually get different chemical forms, polychlorinated biphenyls, brominated flame retardants, the organochlorine pesticides. So this is really something that is now, we're at the cusp of being able to do a lot more in a much more affordable way. And this is also facilitated by availability of chemical libraries. This is incredible how such a simple barrier can exist. A single laboratory, you can't afford to buy 1,000 different individual chemicals and create a chemical library. So now chemical libraries are available in different sources, and this really enables. So I just have a couple more slides, and I'll be done here, Phyllis. So this is the take home right now for where we are. We're in this between 10 and 20,000 chemicals for about $125 a sample. If you add in the GCMS, you can go to about, in that range of 20 to 40,000 chemicals for about $250 a sample. And so that's realistic today in terms of, but you're going to hear more about this later. Major limitation is in chemical identification, because we're measuring all this stuff and we don't know what it is. Now, most of it's not important because most of it's not actually a health concern, and that's where this needs to get into the hands of the epidemiologists to really figure out which of these we should be focused on. So just a couple slides on some key tools. One is MummyChog, which is a program that Shazal Lee developed, and it really transformed everything with regard to this high-resolution data, because it bypassed the barrier that you had to know what a chemical is before you could evaluate it. And so this is a pathway enrichment tool to allow you to be able to go directly into asking question, is this exposure, what metabolic pathways are associated with this chemical exposure? Another tool is one that Tianwei Yu introduced, was a hybrid APL-CMS. For this, you can take any metabolic product. So if you have some environmental chemical and you know what its products are, you can now search you can search the data for those products. And so that's really facilitated things. and a paper that just came out in Analytical Chemistry by Karan et al., which is called XMS Annotator, and this is a multi-tiered probability testing for identification. It's not 100% accurate as far as chemical identification, but it certainly helps you in terms of being able to take things, prioritizing what you want to take to go back and really confirm the identity. So in our workflow, coming back to that chlorophenylacetic acid, very low abundance chemical, we can do a targeted metabolome-wide association study, look at which other chemical features are changing in association with that chlorophenylacetic acid in a population study. We can then come back and look at what metabolic network structures are associated with that, and then put those top features into MummyChog and see which metabolites are actually varying. This is not cause and effect, understand. This is an association. It still requires to go back to an experimental model to see that that there actually is a cause-effect relationship there. And this is work that Doug Walker, he's a PhD student, he should be finishing up next week, defending next week. This is his spaghettigram, and I give this because Chirag Patel will follow with a lot more beautiful figures on this. But this is the problem that you come up with when you start integrating all the omics, as you get, oh, wow, how do you deal with this? What this data showed, this is a study that was done in collaboration with Nat Rothman and Jing Lan at the National Cancer Institute. What this shows is that you can link urinary metabolites of trichloroethylene with immunologic markers that have been linked to the cancers associated with TCE exposure, and metabolically you can link those to renal biomarkers which have been linked to TCE-induced renal toxicity. And so it really shows that bringing back that entire, I guess I don't have this model, it completely shows that you can go from the exposure assessment to measuring the body burden and linking that directly to the metabolic response. The integration, you're going to hear a lot more about this in Chirag's talk, I'm sure, but the reality is there's examples of every one of these now in the literature. We've done a lot of these in our lab, and this is really most powerful for the biological interpretation, is to integrate the exposure information with the other omics methods. So to summarize, where we are now is the ultra-high resolution mass spectrometry with the liquid chromatography routinely measures in that range of 20,000 chemicals. Important thing, as Gwen pointed out, is volume. It only takes 50 microliters for us to do this. For the GC, it takes a little bit more in terms of the sample volume. It takes us about 200 microliters, but it also gives us in that range of 20,000 signals. Again, we don't know what most of them are, but clearly it covers a lot of environmental chemicals. Multiple tools are available, and the feasibility established to use the high-resolution metabolomics to link occupational, behavioral, dietary, and environmental exposures to health outcomes. This is where we are today. This is the future of the exposomes research. And what I've been mainly pushing, so this is just simply editorial, the last conclusion here is purely editorial, is I think that this technology really could be a central reference for personalized medicine and also for sequencing the human exposome. And so for those of you who are interested in the CHIR samples, if you're going in that direction, you have to specifically request. We're part of the CHIR. We have these capabilities, but that's buried among a lot of other stuff. And you have to explicitly ask for this type of information in order to get it. But I would advocate that it's the cost. It's affordable now to basically include this with pretty much any exposome research. And then that way you have a reference to be able to compare to all other exposome research that has been using this technique. Finally, all I do is write grant proposals. These are the folks that do everything. So I already acknowledged many of them, but incredibly talented group. So thank you, and not too much over on time. Yeah.
Thank you very much. This is fantastic. We're going to need your microphone. I think we have time for one question. Yes, Gary Ginsberg.
Yeah, you mentioned.
We're going your microphone. Actually, you provide your affiliation and your.
Yeah, Gary Ginsberg, Connecticut Department of Public Health. So we know that hair could be an integrator of exposure because you could look at the length of it, and I'm just wondering about other compartments that may be available that we may be able to get more than just a snapshot, but look at the exposome. For example, red blood cells have a lifespan. Fat, where could we be getting more history rather than just a snapshot?
Yeah. So I had it in my title, exposure memory. That's the concept of exposure memory.
Exactly.
I omitted. I didn't really talk at all about that but it's very important. There's a lot of ways. Steve Rappaport is working on this adductome concept, the protein modifications. That's very clear. Within the CHEER consortium, there's this measurement at Mount Sinai on the dental and the deciduous teeth, where they're going in and can measure within the different layers of the tooth. So yeah, that is the future. We have to have that. We have a little bit of it, but there's a lot of opportunity. Especially, I think we have, the proteins have different lifespans. So, they have different turnover. And so if you have the adductome that Steve Rappaport is studying, if you get proteins with that have been there different lengths of time, you actually have potentially a way to measure
Hemoglobin addicts, for
Yeah,
example.
yeah. There's a lot of potential there.
Okay. Thank you very much, Dean. Great lecture, and we're moving forward with our next speaker. I actually was so excited about your talk, I forgot to say where your institution is, and I apologize for that. So our next speaker is Chirag Patel, Assistant Professor from the Department of Biomedical Informatics, Harvard Medical School. And Chirag has trained in translational biomedical informatics at Stanford University and has the expertise in the areas of software engineering, genomics, informatics, computer science, and statistics. Chirag published first environmental-wide association study, the WASH, and has been a pioneer in developing computational approaches to capture the interaction of genetics, environment, and health through population-level data. His work involves development of computational approaches which utilizes the wealth of big data available through electronic health medical records, epidemiological cohorts, personal exposure monitor, and genetic information. Chirag's talk today is entitled Challenges and Promises of Building a Search Engine for Exposures in Disease.
Awesome. Thanks so much for that very kind introduction. It's an honor to be here. Let's see, I'll put it down here. I'm all mic'd up, official, ready to go. So I'm a informatician and a genomicist by training. And my key interest, as was so kindly pointed out in that awesome introduction, is using these tools to understand how environmental variation in the world affects phenotypic variation. So I am backed by and honored to work with a group of data scientists with me today. For example, Raj, Chirag, the better we call them, Jake, Nam, and Kajal, who I'll talk about our work today. But our group is a very diverse set of data scientists, mathematicians, physicians, trained in various areas of biology, but mostly with this computational bent. So that's the kind of perspective I'll bring to you today, and I'll highlight these guys as I go on. So as I teach our introduction to genetics class, and as I like to tell our first-year students, phenotypes are a function of genes plus environment, okay? And so phenotypes, what we mean by that, anything from diseases, how genes turn on and off, and the genetics are things passed down from mom and dad. So the millions, essentially 10 million or so variants, common variants that are passed down from mom and dad, as well as environmental exposures, of which that's the name of the game today, and the reason for the season today. Things like the exposome, infectious agents, nutrients, pollutants, drugs, all trying to get a handle on this information is a challenge for us today. So as is alluded to by the great set of speakers, Dean and Gwen alike, we're getting really good at genetics-based investigation. So we've had it already implemented, in effect, a search engine for genetic variants in disease through this paradigm-shifting, very impactful data science exercise called the Genome-Wide Association Study. So here you could peruse if you, at your leisure at this website here, all the results, and in effect connecting millions or so genetic variants that come down from mom and dad to specific phenotypes. So here in this catalog, this Genome-Wide Association catalog, they've assessed on the order of hundreds of different phenotypes, everything from cholesterol levels to disease to gene expression levels. But we just aren't at that level in environmental science today. And so we need to get these high throughput methods to a scalable way, as Dean was saying, to get these data to really do these data science exercises for discovery. And I will argue to you, as I think I'm drinking some of the Kool-Aid, that a similar paradigm for discovery should exist for environmental exposures as it does for genetics. Why is this? Because phenotypic variation, that is disease variation, is a function of genetic and environmental variation and their interactions. I'll leave out those terms. And why is this important? Because human geneticists today compute how much genetic variation is attributable to, or phenotypic variation is attributable to genetic variation with this term called heritability. So it's an indicator of phenotypic differences that is attributed to differences passed down from mom and dad. And so why is this quantity important? Is it gives us a measuring stick, just like attributable risk that you might be familiar with, but it gives us yet another measuring stick grounded in human genetics about the relative contribution of environmental exposures and disease. So here I pulled down some heritability estimates from SNPpedia. These are based on family-based studies where you understand the genetic correlation between individuals, such as twins. And from these studies, you could ascertain that things like eye color, hair curliness, phenotypes are highly heritable, that is, highly genetically controlled. But the reason, again, for the season, the things that we are concerned with today, burdensome diseases today, cancer, type 2 diabetes, heart disease, all have heritabilities less than 50%. What does that mean? That could mean that shared environment, specific environment, and of course stochastic variation plays a role in these phenotypes. So there's a great role of exploration for discovery for environmental variation in the population. And so this is the name of the game here in data science, is trying to explain this hidden phenotypic variation as a function of environmental variation. And we draw inspiration from genomics, so it just took 8 years from going to sequencing of the genome to characterizing how we're different through HapMap projects funded by the NIH and others, to accessible technological platforms to do measurement. So here's just $100 for an assay to do 1 million variants. This will probably be free in the next few years, to doing these data-driven exercises. So this quintessential Nature paper published by the Wellcome Trust Case Control Consortia, where they're looking at 7 common diseases. Now we're up to on the order of thousands of phenotypes in association with genetic variation. So again, as Dean said, the name of the game is bringing the cost down of these measurement tools. And I think this is one of the challenges of exposome research today, is getting the price down of these in a scale and doing these assessments in a scalable way. Here's an example of a GWAS. And what I mean by accessible is that you can really get a large sample size. So this is one of the smaller mega analyses that you'll see in genome-wide association studies today, where they looked at 8,000 diabetics and 39,000 controls in 2012. Now they've scaled up to look at essentially at millions of individuals and phenotypes like BMI, height, and so forth, basically because the technology is so cheap to do measurements. And so here's your negative log 10 p-value of association. You're querying along all the variants along the genome, and here are your top signals for genetic variants associated with type 2 diabetes in a very robust way. These p-values are certainly indicative of something going on there in terms of the genetics, but I showed you earlier, the heritability of type 2 diabetes is quite low, so there's other sources of variation in phenotype that we need to explain. All right, so why do we like genome-wide association studies? It kind of kicked the old hypothesis-driven candidate gene approach to the wayside. Why is this? Not only because the price was the driver, but it happens to be a robust, transparent, comprehensive way of assessing genetics and phenotypes. And this is exactly what you'd like to do with the environmental exposures. It is comprehensive and transparent to your readership. You're assessing all the variants all simultaneously and testing them against a multiplicity control threshold for signal-to-noise ratio. That is, I'm going to say the variants that seem to pop up over that threshold, I'm going to deem for a replication or biological study, and you open yourself up to discovery. And this is one of the main themes of GWAS, is that things that you had never thought of exploring are going to show up in your study to be significant. And these are novel findings, discoveries that you later like to replicate in a follow-up study. And so we've outlined some of the promises and challenges, and this is what this talk is all about. But if you're interested, Raj Manrai with Yusha Kui and David Balshaw led this great review that Gwen mentioned. And we talk about some of these issues today, and also it's summarized by these next couple slides here. That is, high throughput exposures lead to discovery, but I'll also argue that it'll give you a platform for systematic and reproducible association testing, where you can explicitly control for multiple hypotheses and submit things for scientific prioritization to make much better your observational studies, as Dean would like us to do. Secondly, the challenges, though, are we need the standard and scalable technologies. Measurement is there. We can do 200,000 to 800,000 analytes. Now we need to figure out what the heck they are. And so I think that's the next stage of this endeavor, is understanding what we're measuring, because we can measure 800 things at a time. But one of the things I'll talk about today is big data equals big bias. There's things like confounding and reverse causality. How do you handle this in larger dimension? So in genetics, we don't need to worry about this, because genotypes are essentially randomly distributed in the population, and we don't have issues to a large degree of confounding. It's taken care of through population stratification or reverse causality. We also have what we call a dense correlational web, which I'll describe in a second here, kind of alluded to a bit earlier as well. But also we have these fragmented and small associations coming out of these studies. So how do you prioritize these associations that are small and perhaps fragmented? I'll give you examples of this in a second here. And of course, there's the influence of time and life course. So there's, of course, exposures. They change in frequency and trajectory, and they drive the phenotypes in different ways over time. I think Toby will talk a lot about this as well in the next talk. So let me give you some examples of this discovery machinery, or what we like to call EWAS. So until very recently, to the great impactful CHEER program and the Exposome projects that are launched in Europe, there hadn't been many exposome-related data sets. So I kind of cut my cloth on the National Health Nutrition Examination Survey. Already impactful. Every other week you'll see a paper coming out for prevalence estimates like JAMA and New England Journal of Medicine. It's been around since the 1960s, now biannual, and you have about 10,000 participants per survey, a lot of exposure measurements of their targeted nature. You also have a GWAS chip on some of them for measure from 99 to 2000. Also quantitative clinical traits that we physicians like to use, like for diagnosis, like glucose, cholesterol levels, BMI, and of course linkages to cause of death. So, for example, for the environmental and behavioral indicators include drug information, so serum levels of nutrients and vitamins, some urinary pesticides and pollutant levels, urinary plastics and consumables, as well as serum persistent compounds as well. And so what we did was, in our initial studies, is looked at diabetes, as Gwen so kindly mentioned in the get-go, but more recently we've been looking at linking this information to future outcomes, such as indicators related to aging. So we looked at the linked information with mortality, and the way we do this exercise is similar to one you would do with a GWAS. That is, you take your data set, you figure out which ones you're going to use for training. That is, you'll develop your hypotheses in one and try and replicate in another. So we split up the NHANES into 99 to 2001, link them with death information, and assess all the different 246 different indicators with time to death using a Cox proportional hazards model, use a false discovery rate to deem signal from the noise, and then try to replicate in an independent survey data set. So this is what you kind of see when coming out of a a systematic approach. You might see a Manhattan plot, or you might see a volcano plot. So here's a plot of our effect sizes for all-cause mortality on the x-axis. These are adjusted out by age, sex, income, race, all these other things that we hypothesize also impact the exposome, and the negative log 10 p-value on the y-axis. Our signal-to-noise ratio is our false discovery rate. This is something that genomicists have come for granted. Probably the most impactful thing to come out of the statistical literature in discovery research is Benjamini-Hochberg's false discovery rate. And we use the same thing here to deem what is signal and what is noise. And then those that are in dark are the things that are replicated, so factors that we found in the left-out survey. So here's what we found. For example, the things that we used for adjustment that we thought were confounders happen to have very large effects. So, for example, guys relative to females, of course, this is very well known in multiple cohorts around the world. Seems like a biological phenomenon that men die earlier than females. And so if you look in the United States, you have about a twofold greater risk for death. And lower quintiles of income also associated with dying quickly, as well as, of course, age as well. Then you start overlaying some of these exposure factors, so indicators of nutrition, so like cereal-lycopene on the left side of the null, so increased rates of survival for individuals who have higher levels of this nutrient, as well as indicators of smoking and other things of the smoking matrix, if you will, like serum and urine cadmium, which I'll get to in a few slides here. But the top factor here that was associated with all-cause mortality included physical activity. So long story short, all of these different things, all of them heterogeneous, things like smoking status, internal measures of exposures like cadmium and nutrition, as well as behavioral factors like physical activity, all play a role in what we're calling this thing the exposome. How do we all measure this in a systematic battery like we do with GWAS is a major challenge. You put all this into a model and ask how much death you're explaining in the United States in that heritability type of approach. So for example, here, if you ask how much variation in death are we explaining, not too much. So a long way to go for exposome to actually ascertain human death in the U.S. In the future. So great area of opportunity. We took another indicator of human aging, that is telomere length. As you know, as cells divide, as we grow older and older and older, these parts that protect DNA just due to replication, they actually reduce in size. So telomere length is correlated with chronological age, as it's said, but also there are genetic environmental factors that could also influence telomere length. And in a sense, this has been one hypothesis, for example, for cancer. That is to say, perhaps telomeres are longer in clonal cancer cells than in other body cells, just to keep the replication machinery going, or the DNA protected over multiple replication periods. So we did the same kind of approach here. You have longer telomeres on this side, shorter telomeres on this side, connected 452 things with telomere length in the linear regression sense. And you get things like phenotypes like trunk fat, alkaline phosphatase, indicator of inflammation like CRP. Of course, physical fitness comes up again. So indicators of VO2 max associated with longer telomeres, higher pulse rate. Higher pulse rate indicates less physical fit you are, associated with shorter telomeres. So kind of connected phenotypes there, as well as things like nutrient indicators and smoking indicators. So cigarettes per day, cadmium, as was found in the all-cause mortality, seem to have similar connections with telomere length. But an interesting thing here that came up that kind of boggled our mind here, and this is kind of the new, the framework for discovery, is a contaminant was connected with longer telomeres. So what the heck was that all about? And so this is the kind of thing that we need to explain in these future studies, where we get these associations that we need to figure out the biology of. We might have things that we might be in the opposite direction of what we prior hypothesized. So I'll talk a little bit about that connection in a second here. So these effects here on the order of 2 to 8 years, if you're looking at, for example, per standard deviation, the continuous factors of telomere length. But still, we have yet to explain the variation in telomere length after controlling for age, about R squared of 1, when looking at all these 452 factors all simultaneously. All right, so there's a lot of examples of this machinery by us and others in various phenotypes. We haven't gone up to the level of GWAS yet, but we hope with programs like CHIR, for example, we'll get to the childhood outcomes in detail. But if you want more examples, please, you can follow this link here. So I think what the promise is actually understanding the biomedical relevance of findings. And so this is a paper that kind of inspired me during my graduate work by Greg Gibson, who is a professor of biology at Georgia Tech. And if, for example, if we believe that these things are causal, of course, we can't execute a clinical trial, for example, for a lot of these things. So these classic types of experiments on humans are kind of out of it. But I think other ways we could get to this are looking at biological response, for example, in high throughput fashion. So here's one. This is a paper or a perspective actually in Nature Reviews Genetics by Greg, and he says that, you know what, if things are influenced by the environment, you should be able to pick it up in the proteome or the transcriptome. And so here he shows examples. These are transcriptomes of individuals who happen to be HIV positive, HIV negative. So this is kind of the environmental factor here. And what he's trying to show here is that these genes cluster, they're turned on and off, that based on how people are exposed. But there are also patterns that might indicate how one's genome, for example, the group that they might fall in these exposure groups might also induce exposure change as well. So I think if we, by following up in these now commoditized biological experiments, that is, for example, transcriptome or proteome experiments, we may get to the point where we understand if, in fact, these exposures have anything to do with changing biology. Already, if you're wondering where to get some of these data, it's a couple of days after you probably pay your taxes. You know, there's the publicly funded investigations have already been executed for you to download these data today, in fact. So, for example, here's the gene expression omnibus. It has right now on the order of 2 million samples people have deposits because they have to. For example, I'm submitting a gene expression paper. A journal will mandate me to submit my data to the gene expression omnibus. So if you have a quick hypothesis you'd like to test, perhaps the exposures and the genes have already been measured in the gene expression omnibus. So just right now you have on the order of 4,000 different experiments, 2 million different arrays you can download that contain millions of transcripts that are turned on and off. So we wanted to evaluate some of these findings that we found in telomere length and mortality with respect to change in expression. So one example, again, I'm going to bring up GWAS. So here's Cott and colleagues in Nature Genetics in 2010. They showed that genetic variants were associated with telomere length, and they implicated 24 genes in telomere length. Of course, things like TERT, which were well known, TERK, they all are involved in telomere processing, for example. And so we wanted to ask if these genes were differentially expressed when exposed to PCB. So what we did was took arrays from the gene expression omnibus. We harmonized them to the same platform, and we asked, of these genes that had been implicated in genome-wide association studies, were they differentially expressed when exposed to PCBs? And the hand-wavy answer is potentially yes, they were. So here's a volcano plot showing those candidate genes that we pulled from the GWAS literature that were differentially expressed in these PCB experiments. You have things like myoneurin and SLC44 and the solute light chain. Interestingly, these had been also implicated in cancer type of genome-wide association studies. But this was very suggestive, and I think that platforms like this that ascertain how biology changes as a function of exposure with these transcriptome measurements is in fact the future if we're trying to untie causal relationships between exposures and disease. So we thought, let's take a step back and assess feasibility. Listen, physical activity is a huge indicator of chronic disease risk. Let's see if we can find any transcriptome factors associated with physical activity, since it popped up in our EWASs. Others have studied it tremendously. It's been a known risk factor for chronic disease. So Kajal here, a master's student, just finished our Introduction to Data Science course, just downloaded all the physical activity data sets that she could from the Gene Expression Omnibus, and in fact underwent a transcriptome-wide association study in genes that are differentially expressed under aerobic and resistance training, for example. So she took samples that had individuals that underwent physical activity, individuals who were basically under non-physical activity, measured the differences between the gene expression, and executed this TWAS. The thing is, by using a data set or repository like Geo, perhaps like Cheer in the future, you can combine these data sets to add power. And so where one study wasn't powered enough to find an expression signal for physical activity, she combined all these data, amassing on the order of 200 samples, where she found a putative target that I'm not going to tell you the name of because it's still under review, that has some role, we believe, in chronic disease risk. So she found, when she was compiling all these data sets together, she found a spectrum of gene expression hits that were in fact correlative with physical activity and indicators of VO2 max. Just to emphasize the power in numbers game here, so here's the expression for a set of 4 of 11 genes that we found at strong FDR that were highly predictive of physical activity, which were also highly predictive of disease state and other transcriptome experiments that none of them would have passed the bar of strict statistical significance in one alone. So Kajal had to really integrate over multiple data sets to get signal from the noise here. So lessons here is that I think that by combining data sets in this way, by prioritizing factors from these data-driven exercises, we will be able to find discriminatory biological responses and causal factors for diseases that will explain phenotypic variation in the population. I'll argue that this approach is comprehensive, systematic, and we will mitigate false positives. That seems to litter some of these phenotype environmental association literature. I think what their true value add is being able to query biological relationships, such as gene expression, changes in protein. But we do need an expansion, and I think it's up to us in informatics to really make this happen, to query biological roles due to these exposures. Things like Tox21 are just a start. We need things to integrate over the gene expression omnibus that I just talked about, and programs like GTEx, which have been very impactful in looking at variants that are associated with the gene expression measures. All right, so let me talk about challenges. And so I recently, with John Ioannidis, wrote a perspective now 3 years ago about the challenges of studying the elusive environment. I think new genomics technologies to measure exposome. I think Dean and Toby will talk about these. Longitudinal data, this is coming very soon with the programs like Cheer. But of course, to toot our own horn, data science techniques to really understand the connections between these exposures and phenotypes. So again, things that affect our causal estimates or how these things are associated with phenotypic variation includes dense correlations, reverse causality, and of course confounding. And to give you sort of this the spaghetti eye view of things of the exposome, you just take simply the exposures that are measured in a Haynes, which is a very small targeted sample of the millions or so that things that we're exposed to, and already you see this complex web of exposures that exist in, for example, the United States. So here we took 575 factors. This is also Raj Manrai, and we computed the correlations between all of them, essentially 82,000 different correlations. We permuted the data to assess which ones were strong and plotted this in the Circos plot. So all this code is available if you'd like to run with it. But long story short, just by looking at this correlation globe, is you have a sense that a lot of things are correlated with a lot of other things. So that is to say, if I found this factor here associated with your disease of interest, who's to say if it's not really this thing or this thing or this thing that's also correlated with that exposure of interest? So what does this mean in this complex, dense web of exposure? And so we can ask questions once you have this data structure, like which things actually modify these nodes the most. And so, for example, income happens to be one of them. In this paper we published also a couple years ago in AGE, income seems to be a driving force between these hubs of the network, and we showed that, you know, of the exposures that we assessed in Haynes, almost 13% of them were connected with income levels. And here's kind of a snapshot of what these are, things like smoking exposure, as you might expect, air pollution indicators, as well as a whole swath of chronic disease factors. But household and shared environment may also play a significant role. That is to say, exposures that I encounter might be the same as my spouse or my children that live in the same environment as I do. So another question we wanted to ask is, other than, you know, things like income and socioeconomic status, what other things tend to induce, influence, and a variation of exposures? So, for example, you might have individuals who live in the same house. They might be tightly correlated with one another, but they might be more loosely correlated, their exposures with other people in other households. This has huge implications on how we sample the population to assess exposures, as well as routes of exposure and understanding this concept of shared environment. We teamed up with investigators at NICHD, Jermaine Buck-Lewis, as Gwen mentioned. She has this awesome cohort of 501 couples. These couples happen to be living together. So this study was a prospective one to understand the exposures and other factors that lead to fertility and in fact, also sterility. So this provided a great experiment to ask how much was mediated by individuals sharing the same household. And so in this study, Jermaine and folks at NICHD have measured things that you might find in the NHANES, like 36 PCBs, you have polychlorinated chemicals, you have heavy metals, you have things like cotinine, you even have antimicrobials and things that one individual of the house might be using and the other may not. And so already you find things that are reproducible in life that are comparable to NHANES. So again, a dense correlation globe that Jake shows here of the life study, but also the similar patterns, if you will, which I'll try to convince you of in subsequent slides, of exposures in men and females. So the correlations that you might see in males are similar to what you might see in females, for example. However, and this kind of summarizes that as well. So here are Jake binned the different exposures into groups. So you have things that are more persistent on this side and less persistent on the other on this side here. So urinary measured things here and persistent things here. Females and males appear, if you can see the x-axis there, the different categories of exposure, appear to be pretty similar across the board here. Again, this is the absolute value of the Spearman correlation. But then if you ask the simple question, is a male exposure correlated to female, or one person of the couple correlated with the other, you see that these correlations, they dip, they diminish in size. That is to say, while shared environment may play a huge role, that specific environment, the exposures that an individual is exposed to, is different on a daily basis than others. So kind of, it might be an obvious result here, but I don't think it has been documented in such a fashion as Jake has done here. And things like life enable you to do this. For example, one thing that does pop out is, for example, things like cotinine, where perhaps the couples have a high frequency of indeed smoking. So this is one thing that you could see, that shared behavior between individuals living in the same household also could be apparent, but a lot of other things, arguably most things, are in fact not. So again, patterns between co-exposures that I just showed in the correlation globe look the same between males and females, but if you ask the question, how do the correlations shift if you look at males versus females, completely diminish. So these colors here indicate stronger correlations if they're darker, and you can see just qualitatively that they in fact do get lighter as you look at males versus females. So in fact, the exposure patterns are clear in females and males alike, but they diminish when asking if one can predict the other. All right, so it is possible to capture a household-specific versus shared environment using studies like this, like life, and there's a significant role of both. This has implications for how we sample the population and how we assess routes of exposure in this new high-throughput world. So again, dense correlational structure of environmental exposures has analytic and causal implications. We're left with what causes what. The correlations are dense. There's a strong influence of things like income on internal exposures. Shared and non-specific, or so specific and shared environment play a large role, and we need to understand this to understand routes of exposure and covariation of exposures. But the name of the game, I'd ask, is this type of data structure, while it is a spaghetti plot, it may be hard for us to interpret just visually like this. This database allows us to ask, does your new correlation that you found, say you tested beta-carotene in diabetes, does it even matter in the whole space of other correlations? Maybe the effect size of that new thing you found, it doesn't add any sort of R squared, or doesn't compare at all to the whole space of other correlations. And so by having this database, you may be able to prioritize which factors might be, in fact, more risky than others, or explain more variation in the population than others. And so really, how large is this exposome if everything is correlated with one another? Can we figure out ways of like we do in genetics, of summarizing or expanding places in which the correlations are dense and not so dense, respectively? All right, so other things that may hurt us in the future is fragmentation. So here's a great example shown by John Ioannidis and also John Schoenfeld. Here's an example of what they might mean with fragmentation, and this is one issue of looking at things one at a time. So they took 50 random ingredients from the Boston Cooking School cookbook, and they asked, were there any associated with cancer? And here is the relative risk for those 50 the 40 that they chose. So the 50, the 40 were actually studied in cancer risk. So you have things like wine, tomatoes, all the way down to bacon, which all have, looks like from all the studies that they looked at the literature, all have increased risk for cancer, whereas others, they bounce around the null a lot. So this is an example of fragmentation. And what has kind of rampant in this literature is that it's non-replicated, you have inconsistent effects, and non-standardized. So I think what the exposome can allow us to do is, in effect, systematize the analysis methodologies such that we can now are into this regime where we're replicating and getting and having similar ways of analyzing these things so that this doesn't happen. And I'll show you an example of that in a second here. Here's a great clip from our good friend John Oliver talking about that same study. It's a great YouTube video if you want to check this out. So another example of fragmentation are all drugs that we take associated with cancer. So drugs, big part of the exposome. We essentially took the entire population of Sweden that had linked information with pharmaceutical prescriptions, and we asked, did those prescriptions actually predict future cancer risk? So the entire population of Sweden, 9 million individuals, and 500 drugs. And the reason why we chose Sweden is that there's lack of missing information. That is, because of the socialized medicine structure, you have all the diseases that one might have and all the drugs that they're prescribed. And we asked, were there any connected with cancer? And we looked at two modeling techniques. And this is one reason why there could be a fragmented literature of associations, that there's multiple modeling techniques to choose from. So for example, we looked at what drugs were associated with time to cancer. Here's a Manhattan plot of all cancer, breast, prostate, and colon cancer. And what I'm not going to ask you to interpret this, but what I'm going to show you is that the negative log 10 p-values, the strength of the associations here, there are many. Okay, so there's so many too many to be plausible. So you can't tell me that up to 26% of drugs prescribed by physicians in Sweden or in the world are connected with cancer. That's no way that's possible. All right, so this is kind of the big data meets big bias idea. So if you look at the specific cancer types, you have up to 10, 7, and 3% respectively of drugs connected with cancer. And on top of that, you had modest concordance of the tools that you use. Cox proportional hazards versus case crossover. You have 12 of the 141 results that you found in any cancer in one of the analysis methodologies. And you compare that to a case crossover analysis in which you just looked at cases, you only have 12 of those that are consistent in both analyses, all right? So this is the type of issues that we might face when reporting these results, is that, okay, we have a multitude of degrees of freedom in the analysis modalities, multiple data sets to choose from, and numerous sources of biases. So this is a highly probably biased regime what we're dealing with here. Most of these correlations were small, and we probably had bias by indication and other types of confounding bias that were driving up these association statistics to astronomical p-values of 10 times 10 to the negative 100. One way this could happen is the idea called vibration of effects. That is, we have a maze of potential association choices we make, different models, univariate versus adjusted, versus these newfangled machine learning technologies that one might use. All of these lead to potentially different results, and I think it's a challenge for us, all of us in the future, of really understanding how these things work and harmonizing amongst the methods that are possible. In fact, we have a paper that we show how these types of choices alter your inference, and we call it the vibration of effects. And for example, if you're interested, you could check out this website here on tools to compute what we call the vibration of effects. That is, effects due to choices of your model. And these all can highly affect your inference. So again, in conclusion here, I believe that despite the challenges, emerging data science-inspired approaches like the exposome will enable us to discover, okay? So I showed you a couple of examples into linear length and immortality, but I did tell you about challenges such as this dense correlational web, confounding issues, reverse causality, and I think a lot of these issues, if we're properly documented and we can address them, and we can also try and tackle these other issues through study design. But it becomes a problem in how to design these studies, when considering this high dimensionality of these data sets. I think eventually these types of high dimensional data, like it has in genetic studies, will mitigate the fragmented literature of associations that John Oliver has made popular. But I believe that these tools will also enable us to prioritize our epidemiological and biological studies. If you want more challenges, I think we get into even deeper Alice in Wonderland situation when we consider interactions between exposures. For example, mixtures, interactions between gene-environment interactions, and we have a suite of examples and opinions on how to deal with this in the future. That is to say, you have a bunch of exposures here, a bunch of phenotypes. How do you decide which ones to test? How do you prioritize? How do you detect signal from noise in this low-power regime? If you're interested in any of the stuff that I've talked about today, we have an analytics course that you could take online, download our tools that are developed by NAM, go to this website, try and replicate some of our results here. You can grab all the data, grab all the code, and see if it makes sense to you, modify to your heart's content, and publish to your heart's content. If you want ideas, we have some of our data that's, we put it all into one place in this data descriptor. Contact me, feel free, tweet, email. I have a bunch of project ideas. If you want help with the code or help with the data, let me know. I'm also looking for postdocs, a proverbial slide here. So one project that we're working on is predicting HIV prevalence in sub-Saharan Africa. A lot of people, a lot of observational data sets, a lot of tools to build. This is with Eran Ben David at Stanford. Here's some example in bioRxiv of some of the results that we're trying to push this EWAS approach into this new regime of predicting HIV prevalence in sub-Saharan Africa. And happy to state, Gwen just stepped out of the room, but someone other than NIEHS is also interested in exposome-related research. Yes, it's great, it's good, yeah. And other projects that we're interested in are, in fact, can we predict disease as recorded by physicians based on where we live? Here's an NSF-funded project in which we're taking digitalized information by, for example, our physicians that are recorded in databases like the Observational Health Data Science and Informatics technology, for all this health record information directly deposited into these databases where we can now do some experiments in correlating physical environment with disease status. So these data happen to be longitudinal data on millions of patients. They contain diagnoses, prescriptions, lab reports, notes. We're Odyssey, as we like to pronounce it, is sitting there in our IT infrastructure, and we'd like to tap this information to create new hypotheses about disease risk. And so all of this we're trying to make available as well. So Chirag Lakhani, Chirag the Better, my group, is integrating all these data sets together to create this, what we're calling this exposome data warehouse, to integrate with this clinical information to start doing this external exposome type of approaches. If you're interested, please hit us up. And just to remind you, just to set the stage once again, phenotypes, function of genes plus environment, and I believe that high throughput tools will enable us to ascribe to describe the variation that's not described by genetics and that's needed for understanding disease etiology. Again, I'm very thankful to our funders, the NIEHS, NIAID now, and NSF, as well as the BD2K program. Thank my hosts and my awesome group as well. And would love to have your questions now. I'm around for the day as well. So thanks again.
Thank you very much, Chirag. I think we have time for 2 questions, and then we have a coffee break. Questions?
Sorry, I went quickly.
Looks like you're ready for a coffee break.
I tried. I got everyone to go to sleep, huh?
No, you did not.
Maybe you could just comment a little bit, the income.
Yeah.
It's always a little bothersome.
Yeah.
Social, I think everybody can hear me, but
Yeah.
can you just comment a little bit about how our social-political system is involved with this as well?
Absolutely. So I think that that others have, in their documentation of what this exposome thing is, have tried to, call this out. That is to say, if we're trying to be comprehensive in what we're assessing, that trying to also quantify or even measure in our epidemiological studies what this construct is, or develop a construct for it, and use the exposome to, in fact, bring context around that is a priority. I believe it is a priority. I think I showed you preliminary data to hypothesize or to describe that a lot of the variation in these exposures are in fact driven by this very crude measure of social indicators, or SES.
And even in the larger world.
Absolutely. So I hate to I hate to hand wave what your comment is, but I think it's an important part of these investigations is to address this and to find a quantifiable way of measuring this.
Hey, great seeing you again, Chirag.
Hey, nice seeing you, Greg. All right,
Gary.
Gary, what's up?
As a toxicologist, very interested in what you're finding on cadmium and how far you took that, but also what could we be looking at for things like allostatic load? I mean, glucocorticoids, obviously, but are you looking, or are people looking for the exposome to characterize big picture things like stress, allostatic load?
Yeah, I mean, along the same lines as the, trying to capture indicators of SES, I think that is also a gold mine, but I think we need something to seed that those studies to begin with. That is to say, what is our standard now, and can we predict that, and essentially, can we use that predictor to define a new definition of allosteric load? I'll give you an example. Maybe it's a useful one, maybe it's not. So one example that is getting very popular in the epigenetics community is developing a indicator, a machine learning predictor of chronological age. So Steve Horvath and colleagues at UCLA, they essentially did what I was talking about with GEO, is they just took all the publicly available data sets in GEO that happened to have been annotated with age of the sample, and they built a simple one-line machine learning thing, a linear regression, and they saw that these epigeneal marks were strongly correlated with chronological age. So once you have that correlational machinery, that predictor, you can then start asking, what are the deviations from chronological age, right? So if you have a predictor that's saying this person is in fact age 50 when they're really age 30, that might be indicative of something going on in their endogenous something or another that's indicating their age acceleration. So what I'd say is that can we build something like that for allosteric load? Can we build a predictor using these new high throughput phenotypic measurements of allosteric load? So I hope that example was informative. It's one that came to mind. If you're interested in papers, I could send them.
Okay, well, we have 15 minutes for a coffee break. Before we go to coffee break, I would like to tell you that Caroline Johnson will chair the second session. Also, I would like to thank one more time Waters for sponsoring this event, and we have a table with Waters people outside. So if you would like to meet with them, if you have any questions regarding the hardware on mass specs here. Also, Dr. Collman and Dr. Jones might depart a little bit earlier, might not be able to be around on the round table, so it's a good opportunity. If you have questions for them, grab them at the coffee.
Hi, so welcome back everybody. It's my pleasure to introduce to you Dr. Toby Athersuch, lecturer in environmental toxicology and biomarkers at Imperial College London, UK. His primary research focus is on the development and application of analytical techniques such as high-resolution nuclear magnetic resonance spectroscopy and mass spectrometry for metabolic profiling. He has worked on several key molecular epidemiological studies and is now involved in developing the concept of the exposome through the incorporation of metabolite analysis and has been involved in the EU-funded Helix and Exposomics initiatives. The title of his talk today is Oscillating Wildly, Characterizing Metabolic Phenotypic Variability in the Human Exposome.
Great, thank you very much. Thanks, Caroline. Okay, and yes, well, thank you very much for inviting me here to this beautiful place. And hello to everybody on the internet, wherever you are, all furiously logging in, I'm sure. So the title of my talk is related to the title of the Smith song, but that's actually more relevant to the fact that one of the things we are really interested in understanding is metabolic variability, particularly on the individual level, because hopefully it will help us understand and model some of the other things we've heard about in relation to the human exposome this morning. I've got a bit of a difficult job, I feel, to follow Gwen, Dean and Chirag, as I'm sure you'll agree their talks have sort of blown my hair back a bit and got lots of fresh ideas rushing in there. But for my part, I'm going to hopefully take a bit of a different tack. My background is it's actually in drug metabolism, and one of the things I'm interested in here is where we've kind of come around through drug metabolism. I've been involved in population studies as well, but now bringing all of these things together in terms of the human exposome. But for several years now, I've been involved in collaborative projects which have ultimately sought to link things we can identify in the environment with health outcomes, and obviously something to do about that, so the policy in action. But probably I may have a slightly different set of experiences to many people here within maybe the School of Public Health, in that maybe a different range of experiences. I've not been involved intimately in all of these aspects, but definitely been trying to establish in some ways or contribute to projects where we've looked at various different levels of molecular complexity. I guess it would be one way to explore that, right the way down from sort of individual molecule level interactions right up through to trying to understand some of the other sorts of things we've been discussing in this afternoon's talks related to large-scale epidemiological studies and exposures in the environment. And I think one important thing we should remember when we're doing all of this is that one comment earlier was that there's been a lot of work done in epidemiology, particularly about correlative analysis in terms of linkage between exposures we can find in the environment and within people and disease outcomes. But actually we shouldn't throw away a lot of the other information that we can gain. And ultimately the end of this is that although we can find out that we have these associations between environment and health outcomes, is that we need to know why any of those things are actually occurring. And so I think it's one of the jobs that maybe I have and the group that I'm working is with, we can start to try and address that. And hopefully one of the aspects which I'll draw out later in the talk is related to looking at this one aspect of it, which is individual metabolic variation. So I don't really need to explain what the exposome is, but for anybody who wasn't here or was asleep, that we can obviously think of our totality of our lifetime exposures as actually being quite a complicated brew of things. Just going through here on this kind of cartoon, right at the start there, we might have exposures associated with exposures in utero, which might only, well, obviously only last for that period of time. That might include something like smoking. And then we might have other things which are cumulative exposures over a whole lifetime, which might include, say, heavy metal exposure, sort of chronic exposure to that. And then other kinds of exposure patterns which we might find for only periods, maybe an occupational exposure that only happened during the working lifetime, and other things like maybe exposures to individual small, short and idiosyncratic periods, maybe exposure to particular drugs or so on. And then other things which might sort of oscillate with some sort of periodicity, maybe annual or weekly or what have you, maybe something to do with allergens. So we've got all these different types of exposures, and clearly the responses we elicit from those will be related to those same sort of patterns, and they occur within, as we've heard, these what you might define as critical periods. And the thing I'm going to concentrate on later on is this critical period, or these ones earlier in life. As we've heard, that obviously if you step, these early lifetime exposures can potentially be, there can be a potentially critical period for exposures. I'll leave it at that. So I'm interested in metabolite analysis, and we've heard particularly from Dean that we can get fantastic instrumentation that can characterize biological and chemical mixtures in exquisite detail, and we can employ these in a variety of different contexts, I guess, in terms of environmental health research. So on the one hand, we might consider parts of these studies to be biomarker-led, i.e. things we can measure and identify and know what they are. Over on that side, related to the sorts of exposures that we might have. And over here we have other traditional measures about measuring those same, well, some of those same types of exposures. Maybe they're to do with questionnaire data or environmental exposure models. And these analytical techniques we can employ, we might actually employ them for several different interlinking types of analysis. For example, we might just simply look at compositional analysis, as has been done in several studies, and some of you may have seen some of the work that's come out of the group of which I'm a part recently, looking at nutritional exposures and urinary signatures. So compositional analysis is literally using those same tools to look at what's going in in the same way as looking in biological samples collected from individuals in population intervention studies, whatever you like, to maybe look for those same things or characterize those same individuals. So we've got another set of exposures, another set of ways in which we might use metabolite analysis. Might be, as we've seen a long time ago, really to do with case control analysis in terms of diagnosis of disease. And I think that's something we haven't really mentioned a lot today, but actually has a lot of benefit here, because understanding the metabolic perturbations that relate to diagnosis, i.e. once we have pathology, obviously we don't want to get those confounding our analysis when we're looking for other things which we might consider to be predictive of disease. Anyway, we can combine these things in lots of different ways. Maybe we might look at these exposure markers that we've explored through composition analysis. We might combine those with the composition analysis with looking for biomarkers of exposure and get exposure biomarkers. Or we might look for the response and compare those to these components which we've been exposed to and get response biomarkers. And something that's not really been put forward so much, as far as I can work out this afternoon, is that a lot of the work that's been done in the exposome is relying on these prospectively collected nested case control studies, where, as many of you will know, we have biospecimens collected many years before we are now doing the analysis, and we're able to select from those biospecimens only those which we wish to analyse based on, say, the case control status. And that allows us to go in and look in those prospectively collected samples for measurements which may be predictive of the subsequent outcome. And so some of the early ideas about how we were actually going to do this exposome in a sort of functional reality was, on the one hand, if we were able to do that with these prospectively collected nested case control studies, and link those to endpoints, we would end up with intermediate biomarkers of early effect, i.e. predictive markers that tell us about something before the onset of disease. But also, on the other hand, we might end up with either things we knew about already, but maybe new markers of exposure. So effectively reaching out on one side to exposures, and on the other hand to prediction of disease. And this whole idea was sort of termed as a meet-in-the-middle concept. And it's got its limitations, but it's one of the things that sort of helped gather some momentum around this idea of using omics markers, particularly for these intermediate biomarkers of early effect that could help match these two things together. And the reason we wanted to be doing that is because we actually want to know what the mechanisms are that link people's exposures to things and what they actually do to why they get sick. Ultimately, if we only ever have associative studies, then we're no further forward in many ways. So those who have exposure to sort of toxicological literature and regulatory toxicology and things like that many others may have come across this idea of having adverse outcome pathways as a kind of natural development of the way in which people are seeking to put together different bits of toxicological information to help us understand mechanistic linkage between exposures to toxicants and disease outcomes or pathological outcomes. And so a sort of most basic version of this model we might have where we're able to characterize the exposures, as we've heard earlier, maybe individual exposures. We know a lot about those. We might work through looking about how they particularly bind to individual receptors or proteins or what have you, and then look for cellular responses and build up through organ responses and so on, through to population responses. And I think as we think about, now we've got this sort of concept of the exposome in our heads, and we move to ever more complicated exposures, we're wanting to maybe consider the contributions of complex mixtures of exposures in the environment on top of all our dietary and other kinds of internal chemical exposures that we have. That this idea of developing more computable network models that help us understand how particular types of initiating events, which may be caused by many different types of chemical, how those then link through to cellular organ and later on to population responses. So I think actually this paper was released a few years ago. It's actually does quite a nice job at it, trying to help anneal the ideas of the exposome and these adverse outcome pathways. And AOPs have their limitations and some detractors in the community, but I think if we're looking for some sort of way of saying we want to include formally some sort of way of including mechanistic information into these exposome models, then this would be a good thing for us to include. And I was going to mention something else there, but I've forgotten. I'll come back. Okay, so just going to quickly move through this. We already spoke earlier with Chirag, gave some good examples about genetic risk, and obviously we're getting ever better at explaining and subtyping people in terms of genetic risk. But we also have more sophisticated ways of thinking about how those diseases are manifest. So some nice analysis here reproduced from this paper now. Again, I can't believe these are so long ago, but they're I think they're very worth bringing up here. In terms of how we might link metabolic genes and metabolic enzymes and metabolic network into converting things with diseases associated with deficiencies in those genes. And the take-home message here is that really that you can find particular clusters of diseases all have similar connections to multiple metabolic components. And so what we end up with is that we have effectively a very complex set of disease outcomes which are actually very highly linked on a mechanistic basis, which is why we end up seeing in many cases that we end up with multi-morbidity. Well, anyway, in this paper there's some comments here, and I think where's the one I was going to say? So the one that I think is most interesting, however, is increasingly understood that such an approach, this is the gene-based approach for identifying these things, is successful, but unfortunately most cellular components exert their function through intricate networks of regulating metabolic and protein interactions. And I think we can all agree, looking at the maps and things we saw earlier presented by Jean, we have these very complex metabolic, even just metabolic networks that interlink a lot of the things. So it's no surprise that actually thinking less about specific disease endpoints and more about those nodes which might represent those parts of metabolism particularly that help link together different types of disease states. So what's this guy rambling on about? Okay, so on the one hand we have the idea of AOPs being potentially useful in that they help us formally identify mechanistic pathways that link molecular initiating events, i.e. many things could cause this event, but once it happens then we can then say, okay, we're now likely to see a set of other events. And then at the other end of this potentially very complex modeled network, we might find that actually we find perturbations in that network that link out to many different disease outcomes. So instead of just thinking about one disease at one time, we might be starting to think about these disease clusters which are related to particular nodes. So I think that some of the comments earlier were useful, were not, they painted quite a dire picture that we're going to have so many exposures on one hand that we can't possibly look at all of these and how they all affect everything. Well, actually, the AOP model might help us minimize the amount of effort we need to put in once we identify common causal pathways, that once we have these molecular initiating events, we can then sort of effectively ... get to the end and then use this at the other end to help us understand the interaction of different diseases. So this whole idea of multimorbidity, two or more concurrent diseases. So what do we want to say about this? This is basically the fact we get linkage because multiple diseases have common biological networks that underpin them. And I think one good example of this, and there was a gentleman earlier who mentioned allostatic load earlier on, i.e. the ability for an organism to adapt through changing. I can't get into all the details of this, but I think this is an absolutely fantastic piece of work. And this group had collected, they only had a small number of individuals involved in the study, but they actually had samples repeatedly collected over 10 years, I believe it was. And they found during that time, those multiple collections, as you looked across that, well, actually people kept a very strong core metabolic component of their metabolic phenotype that had been measured. And this figure here, it's illustrated here in these prediction accuracy, how well one of your sets of measurements could predict your metabolic phenotype of another set of measurements. And in fact, Dean showed a very similar example to this earlier on, a Johnson 2010 paper, if I remember rightly. Anyway, what's the take-home message about this is that actually that these people, including some in here, they indicate some people who had been on antibiotics, so some of them predictions went a bit wrong for a bit, but then they kind of recovered. So there's an idea that we have a sort of an elasticity and ability to respond and then come back to some sort of equilibrium or normal position. And so thinking about how we're going to compute the utility of all of this metabolic and other information we have about these biological networks, actually thinking about this, I think this is really, really the way to go, because what we can define from these kinds of things, potentially, is that individuals inhabit their own kind of biological space described by these networks. And as we've seen, we can identify particular events which perturb that network. And if it's perturbed in a particularly substantial way, we might find that people, the consequence of that is they divert from their sort of metabolic or biological normality, but are unable to return to normal, which might give us reason to imagine that they're then going to be susceptible to subsequent effects, disease, and so on. So that's my kind of way of thinking about how I would try to encompass these different ideas, and I think this is a fantastic example, and the one that Dean gave earlier was also really useful. Sure, I'd put up this as from a while back, but it's all good stuff, is on top of all of this, we're not thinking about this idea of pathways, that this goes to being this, and then it goes to doing this, and then that's what happens to these things. If you've done drug metabolism, you'll know that variation in between even very similarly housed animals, although similar, you'll get differences in the outcome, the proportions of metabolites of those drugs. And that's because we're not talking about a simple procedural way of handling metabolism. We effectively have a sort of probabilistic model, and every one of those events we have in the body is going to be effectively governed by some sort of probabilistic event. So lots of methods, and this is not my area of expertise, but my understanding is that lots of methods to do with Bayesian statistical methods about how we can describe a distribution, both going in and coming out of particular models, might be a useful way of doing that. And if you think about how we might apply that to those networks, and how when we have a particular perturbation, we could describe at the other end some kind of distribution of variation that we might expect from that. That might help us. So this is just really a reminder that we're not talking about sort of a matter of fact, A goes to B. Here we're talking about probabilistic and potentially multiple splitting pathways for lots of different aspects of biology, particularly in terms of metabolism. So the reason I'm interested in thinking about that is because if we're to embrace these kinds of models, think about probabilistic models, or even if we're not interested in that, we just want to know about how these networks behave, when they're perturbed, then obviously understanding what they're like normally in the normal run-of-the-mill things, how much variation they have in the normal run-of-the-mill, is something we want to get at. Because if we know the variation of effectively the prior that we put into some of these models, if we're able to reduce the uncertainty in those sorts of measurements, then that helps us build a better model and helps us do better predictions, particularly when they're very complicated models. And again, it's not my area, so I'll divert onto something a little different now. So how we'll actually get at this, looking at variation, because we saw earlier that people collected samples over 10 years. We can't necessarily be very productive if I just start a load of collections now and wait 10 years from now. Fortunately, we have several collections that have been started, so thank you to all the people who had the foresight to do that several years ago. We're now reaping the benefits of that. But in terms of these longitudinal collections, which is really what we need to establish the variation we see over time versus of an individual versus the variation we see between individuals, obviously these longitudinally collected sample sets are something that are very precious to this kind of research. Obviously in adult studies we can look at all those things I've just described. Particularly interesting when we look in mother-child cohorts for describing these early life exposures. And obviously we want blood and urine for these for us to be able to measure both exposures and omics analysis. And some of these longitudinal and cross-sectional studies, for that matter, studies have done before, investigating the impact of anthropometric factors, so we have some interest, some understanding of the variation, normal variation we see in adults. There's less so, particularly for untargeted metabolite analysis, that has focused on children and adolescents. Some of these studies that have been previously looked at have described this sort of proportion of variation that can be explained, broken down into different contributing factors, for example, environment versus genetics. But as I mentioned before, we're really interested in doing this, particularly in child studies, because of this being really a critical window. So understanding the normal variation in that period versus variation that appears to be related to particular exposures is something we want to look at. So there we go. I've laid out my case for why we want to do that, and as luck would have it, literally today this came out from our lab, so thank you. And so this piece of work is just a preliminary piece of analytical work, really, for us to do exactly that within the context of the Helix study. So at Imperial, the PI for the Helix project is Martin Cohen. Martin at Creal in Barcelona heads up the whole project, and I think it'd be useful for me to firstly explain what we were trying to do and then put it in the context of the whole project. So one of the things we wanted to do, both to give advice for other studies but also for us to think about how we're doing our work, was to determine as best we could the best time to sample these children, whether you do it in the morning or the night, or whether you pool morning and night urines, or what you do. So that was our relatively simple aim. And from within this whole larger study, we had 20 children who provided daily urine collections for 1 week, giving spot urine in the morning and the evening. And we also made a physical combination of those two in those pairs where they were available for us to look at an actual physical pool between those two as well. So the project, so I'll come back to what we actually found in a minute, but just to divert to explain a little bit about Helix, it was kindly introduced earlier. This is one of the 3 projects that were funded through the FP7 Exposome project, Exposome consortium within the EU. And the idea is that it's going to focus on early life and explore exposures to environmental pollutants with child health outcomes. And it relies on, well, there's the paper if you want to go and read it, everybody and their wife on the list of authors there, but it's a huge, great project. I mean, these exposome things, as you're getting the idea, we need people doing all of those different kinds of things. Exposure analysis, people doing the actual sampling, doing the epidemiology, personal monitoring, all the rest of it. So there's a whole load of things going on there. But this one is focused on 6 cohorts, mother-child cohorts around Europe. And the one that we're interested in for the topic here, just so you know, is INMA, which is based in Spain. And so from each of these, so the whole enrollment in the study is 32,000 individuals or participants in those studies. But the children who are in each of those things, you can see their age ranges at the time of the study getting going. And here's an outline of, this is from the paper, so again, you can have a look at this, but I just want to outline the main things. We have the entire cohort, of which there's obviously other measurements, so about those very broad measurements of those, of which a sub-cohort has been selected. So 1,200 mother-child pairs are taken there. Within that, we also have 2 panel studies. A child panel study, which is looking at a subset of the children from each of these cohorts. And the idea of this is to, and the same for the pregnancy panel, which is looking at the mothers. So the idea of these panel studies is to look at uncertainty, variability, behaviour, blah, 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 blah. These things that I'm saying are very, very important for us to look at and identify things we want to either control or understand in the context of the sub-cohort study. As you probably will have seen on here, we haven't mentioned much of it today because the focus has been on sort of biological monitoring and metabolomics and all the rest of it. But actually, a big part of the exposome, the way it's been sort of functionally implemented, has been to try and include high-resolution ambient sensors, either personal monitoring or in the home or these kinds of things, and obviously combining that with all of the traditional types of epidemiological tools that are available. So there are a lot of other sort of measures, activity data from smartphones and things to do with these panel studies, so we can get a really good focused look on many different bits. I'm only going to describe one bit. So once we put all this together, we're going to have omics, we're going to have exposures, and we're going to have all this kind of real-time activity and monitoring data. We're going to do all the number crunching on that and look at how we can influence health impact assessment. And that's the idea, really looking forward to sort of Europe-wide ways of improving estimates for burden of disease and so on. So there we go. So that's where we're in, and just a quick run through for those who are interested in. These are the individual measures which and where they're measured. These are done by a variety of different groups in the project, either looking at things in blood or doing things to do with indoor air or water. And you can see there's a whole bunch of things which we haven't got time to look at here. You just see that all of these things are being done in this Helix subcohort, okay? Which a small, a subset of those are being done within the child panel study specifically. Similarly, there's sort of external exposure measurements, models being made for the whole lot. And as you can see down here, looking at ambient air, noise, UV temperature, and things to do with sort of environment like green space and how they feel about where they live, urban greening and things like that. So there's different types of these external measurements, and obviously the thing I'm interested in is all the squidgy stuff, which is all the omics. My interest is in metabolomics, which is what we've been doing, and the thing I want to concentrate here is the proton NMR spectroscopy. And that's been looking at, I'll just skip to this one so you can see, it's in the child panel study, and the main thing was really looking at variability measure. So hopefully we're clear. Why am I doing all this? It's because I want to get a better estimate of the normal variation in some of these types of measures, particularly in children, because that's where it's a critical period of life. And it's really important for us to know that in the context of this study, because we're putting all of that stuff together. So we have really vague estimates of what's normal for individuals here, then we'll be potentially at a loss. Okay, so what do we do? So we use the protocol, which many of you will be familiar with. if you do any kind of metabolomics. Basically, 1D proton NMR spectroscopy at 600 megahertz is kind of the standard thing that most people do, and it's very robust and reproducible, and this paper just shows you that you can basically get the same answer out of different spectrometers and reruns of the same thing. It's all very nice. So we're able to apply that, and there's the details. Basically, proton NMR spectroscopy, standard sets of acquisition parameters. And one thing we did here was, due to some of the analytical or physical properties of atomic nuclei versus the protocol we used, we did some additional measurements to help us improve the quantitation accuracy of some of the metabolites we've measured. But here we only took a small number of things you can measure with NMR. We ended up with 44 metabolites, which we took as a sort of set that we were going to take forward. We could identify for the purposes of looking at different, breaking down these different types of variation here. And those things were identified via a bunch of different reasons. Those of you who are interested, that's what an NMR spectrum looks like of some urine in the morning, in the night, and then a pool, which is just the morning and the night, 50-50. As you imagine, that looks very much like both of those put together, as it should. And the idea here is obviously we can identify peaks here that are unique to individual metabolites, and we integrate the region there, take those integrated regions out, and we use that effectively as a proxy for concentration, or we can actually calculate the actual concentrations of those things, which is what we did for most of the cases where it was possible to do that. Okay, so we looked at the analytical variability. So this is the looking at effectively a pooled QC sample, looking at variation, which was relatively low, apart from things which had a very low signal to noise. So this was an interesting thing that Dean pointed out earlier, that so you need to, maybe not for NMR, but for mass spectrometry, things that are down there in the grass, maybe picking out what things are real and which things aren't, is something that we should be very, very cautious about when we're trying to add any sort of analytical certainty to when we're reporting them. So some things were very small, which weren't particularly useful. What we wanted to do in this study, if you remember, was to assess variation that people experience within themselves, or within their urine profiles experience over time, which might be a combination of different types of cycles and variation within them, versus the variation you see between different people. And so one way of establishing that is this intraclass correlation coefficient, ICC. So basically, it's this, basically just comparing the different sources of variation. In this context, higher ICC for a particular metabolite means it's more stable in the profile for an individual over time. So we can start looking at that. If you just look at how those ICCs for the things we looked at were distributed, you can find that we find that sorry, what am I saying here? So you can see that there's different distributions for the three different types of sample, and the night slightly lower here in terms of its distribution, whereas the daily pool slightly higher up in this curve, just kind of getting a nod ahead there to suggest that it gives a better representation of the stability of individuals' profile over time. But for individual metabolites, you can see here the morning, night, and pool. That's not come out very well there, sorry. But basically, the pool it has a higher ICC than all the others for most of those things we analysed. So the take-home message there is that really we want to use this daily pool if we can, because it improves our ability to look at the stability of individuals over time. Also used a mixed model to look at decomposing the variation we saw in each of these metabolites. Again, this subset of all the things you could measure. These are all most of the big things you can see in a spectrum quite easily. And you can see some things are dominated by individual. So the variation is dominated by the variation within an individual child, whereas some things down here are dominated by variation that we see between days and technical variability. So things up here, if you remember before, there was a certain that study where they had those things over 10 years. There's a sort of set of core things which are likely to not really be changing a whole lot because it describes you and the things you do and your variation and the ways all your dials have been turned. But down here we might find things that are really fluctuate a lot due to things potentially like diet or one-off exposures to pharmaceuticals or whatever. Injury, inflammation, lots of things. And some things that obviously are influenced quite a lot by diurnal cycles, which we can see a couple here where we do see that taking a bit of a role. So it's interesting, and this will help us inform our subsequent analysis. So it's not potentially for many of you who aren't going to be doing that analysis, nothing to write home about, but the point is that it really talks to this idea we want to, for specific population, we can't just treat everybody the same for starters, we want to look individually at children to try and pin down the variability that we see for them. So there's a summary which I've kind of described all of this, just a summary of the different types of metabolites which we saw which show the greatest inter-individual variability. Those are the things at the top of the plot and things down the bottom of the plot which were not. So our suggestion here is if we can, to best capture the individual variability for these models, then we would use pooled day samples if we could. And just pop this in, this isn't the same study, this isn't in this paper, we haven't done anything with this, it's just some preliminary analysis of where we're going with this, and I think this is where we're going to have to start picking your brains, Chirag, about how we do more clever things with some of the data that we've got. Down here are all the types of exposures that have been measured for biomarkers of exposure within this panel study, and across over here all these different metabolites that we've got, and you can see positive or negative correlations with some of those. And let's just go in and pick a couple. So we're looking at correlations here with metals versus trimethylamine. Trimethylamine N oxide, which is a mammalian and gut microbial combined product, or something you get from eating fish. So, interesting there, heavy metals may relate to fish intake or something that's perturbing the gut microflora. So this is a story which we'd like to unpick later on. I'm not putting anything onto this yet, just to show you this is the next sort of step in terms of the analysis. We've got a good look at the individual variation. Obviously, the next thing we want to do is correlate the exposures we see with the metabolome that we see in here. And here we're using urine. So we've talked a lot today about measuring in biosamples. So urine samples, that's all very well. I would say for the majority of people doing the exposome projects, they're really focused, or metabolomic stuff within the exposome projects, they're going to be focusing on serum or plasma, because I think it's more representative of the internal chemical milieu. But they obviously do different things. Like urine is pretty good at telling you about obviously all those metabolites, different things which you've excreted. So that might help us add a different flavour to the analysis we do. Anyway, that's what we decided to do, so we're sticking with it for now. Next up, this is a relatively recent paper from the folks at the National Phenome Centre, which I'll just mention in brief in a minute. Next sort of thing is to do UPLCMS, like QTOF analysis of the same sets of samples, and we're also going to be doing targeted analysis by the Biocrates kit, which is, that's quite a useful way of bridging the gap between people like me who like numbers and chemicals and epidemiologists who like things that they can do their epidemiological analysis on. So it's kind of a good way of giving immediate, getting us to talk to each other quite rapidly with that targeted analysis. I'm just going to finish up now if I've got just one more minute. In case anybody's not aware, there was some discussion earlier about sort of US resources for high throughput metabolite phenotyping. Similar sort of idea here. The National Phenome Centre, which is a collaboration, funded jointly by the MRC and the NIHR, and based at Imperial, although there's now one also at Birmingham, and they're setting up other instances of this Phenome Centre actually around the world. So that's quite good. And the point was really to combine NMR, mass spectrometric platforms, and the informatics, which we've got into one place to do things in a pretty big way. So the focus here was in a different kind of tack to what Dean described earlier about having very high resolution mass spectrometer. Here we're getting very high UPLC, but with many different parallel platforms that deliver lots of different mass spectrometric assays in parallel. So there's all the stuff. But if I just show you here, these slides kindly from Matt Lewis, who's the director of the analytics there. They've got a whole bunch of QTOFs all set up in parallel with the same sort of, with reverse phase chromatography and hydrophobic interaction chromatography for doing things that are hydrophobic and hydrophilic respectively, and then those divided up into things where they can just look at lipids, really for blood analysis, and then blood and urine analysis, and then things for doing different things by HILIC, and then bespoke instruments for doing in positive and negative ionisation mode. So here's a kind of like massively parallel type of analytical platform for doing the same sorts of things. And the benefit they have from this is that dedicated instrumentation and dedicated pipelines for each of those things is going to be very beneficial in terms of uptime and also throughput and ability to incorporate new, basically by extending out this set of instruments and to including targeted analysis, of which many are now available. So, we've discussed this, which some do blood and some do blood and urine. And really the plan for these types of analysis is that over time, introducing other types of matrices, those matrices, the sample preparation will be tailored to fit the matrix to the analytical platform, which will benefit all of the databasing and the work pipelines that they have there. So again, I didn't want to make this into an advert, but I just thought you should be aware of the fact this is happening. We've had all this discussion about standardization and people talking to each other. So I'm telling you that there's a huge analytical center there doing good stuff. Okay, so to finish up, why we're doing all this stuff, because we want to populate these network models, really, that will help us understand the mechanistic linkage between exposures and disease outcomes. And I think, for my money, the AOP's kind of formalism will be quite useful in doing that. Hopefully it will reduce on the one end the fact we've got lots and lots of different exposures, many of them all at the same time. And at the other end, we've got lots of different diseases that are all actually linked by underlying gene and metabolic networks and so on. So that would be nice. So we want to use these sort of systems toxicology approaches for looking at that. One thing we need to characterize is the, in order to understand the uncertainty in those models and the measurements we make and the context for the measurements we make in exposome studies, is to understand what normal variation and individual variation looks like. So that's why I kind of gave you the stuff we've been doing within Helix. And obviously that's nested, that study's nested within the wider Helix study, which will be combining these omics measures, exposure measures, and personal monitoring ambient measures for child exposures. So, my final slide is to say, what do I actually want? So I think many of us have said this today, is longitudinal studies. It's not really my specialty to set up cohorts, but I think the fact that people are getting the idea we do need longitudinally collected samples, that would be absolutely fantastic, because that will help us deconvolve short from long-term exposures, enable us to understand the sort of dynamism in metabolome and other types of omics measurements. Characterise this individual metabolic variation, that's absolutely critical. Some others have mentioned about, Chirag particularly, saying about if we can actually make all these data talk to each other and just putting things together and just making that N number absolutely enormous. I think if we'd have all agreed in the metabolomics community 10 years ago, irrespective of what it was, to do one assay, all of the same, then potentially we would end up in a better position than we maybe are now. And I think that it's a difficult balance between technological advancement and the academic academic freedom to want to do something that was better than the last thing, versus the ability to do things with other people who you may or may not agree with whether or not they're doing the right thing. But we all kind of have to bite the bullet at some point and make this. So hopefully through the Exposome cores and the phenome centres and things like that will emerge these kind of common protocols and common practices that I think will really be most useful. So I think with that, potentially one way of this is great for everybody to be doing this and knowing how to do it, but actually these centres represent something which have a very low barrier to entry for individual researchers to be able to get information and data from them. They don't have to invest in all the infrastructure and all the rest of it for these large-scale epidemiological projects. So I think, for my money, that would be the way to go. So apologies that I may have overrun a little bit, but I just thought I wanted to put in those extra things to tell you what I want, because that's what we need. There's many people at Imperial who support me in both analytical and epidemiological departments. These are the people who worked on the metabonomics things for the Helix project, and then the Phenome Centre, loads of them there, and many other people who work in lots of parts of the Helix project, and all the other people who support me and also our department in also the work we do. So with that, I'd like to thank the organisers again for inviting me to this fabulous meeting. I've learned tons already, so I just really want to speak to you all later over some drinks, and I'll shut up. Thank you.
Fantastic talk, and I really liked your summary at the end as well. I think in the interest of time, we might move on to the next speaker and save the questions for the roundtable. Is that okay? So it's my pleasure to introduce David Grant. David Grant is Professor in the Department of Pharmaceutical Sciences at the University of Connecticut, and he's also the recent director of the Waters Corporation Laboratory for Biological Mass Spectrometry at the University of Connecticut. The long-term objective of the research in Dr. Grant's lab is to develop analytical, computational, and database tools that can be used to rapidly identify the chemical structure of compounds in human biofluids. These analytical and computational tools will be useful for metabolomic applications, including understanding disease mechanisms, enhancing the speed of disease diagnosis, and enhancing the accuracy of disease prognosis. And the title for his talk today is High Throughput Structure Identification for Metabolome and Exposome Analysis. How can we get there?
Okay, how's volume back in the back? Good, okay. Thank you, Carolyn, for the invitation. Bacillus is not here, but thank you. And thanks for staying around till the end. Appreciate that. I won't probably go over, so. My lab focuses on basically one problem, and that's structure identification, and we have one instrument. So we're pretty focused on doing one thing. So our chemical space is not so big. We heard earlier about people looking at 200,000 compounds or so. I'll talk a little bit about how our chemical space is much, much smaller to try and address this problem of structure identification. So we simplify things quite a bit. Okay, challenges that exist in this field. I think the previous speakers have done a really nice job in addressing some of these challenges. Toby just talked about variability of metabolite levels in humans. That is a big problem. A lot of people don't appreciate that as much as they should. I'll just show you one example of that. It's not something that's easily overcome unless you have a huge population size. The second problem, oh, so they can hear us? Okay. They can see you. Okay. Okay. The second problem is the major focus of my talk, and that's to try and identify structures. Not just the feature, but the actual structure of these unknowns. Okay, here's an example of the first problem. So this shows a graph. On the x-axis is time of day. It's a 24-hour time period. The y-axis is peak height, which is basically the amount. They're looking at one chemical, and that's acetylcholine, and they're looking at that in plasma. They have 8 different individuals. Each line is a separate person. Okay? These individuals were maintained under controlled light-dark, sleep-wake, posture, caloric intake. And you can see with just these 8 people the fairly large variability between the people and over the course of a 24-hour time period. Now, I'm not suggesting that this is the case with every metabolite, but I'm suggesting that unless you know this is not the case, it's difficult to make conclusions about sampling and what it means. Okay, the second problem is the one I'll focus on for the rest of the talk, and that's how do you identify the structure of these features when you have so many to choose from and you have so many isotopes, structural isotopes. Okay, so a typical problem in metabolomics, very, very simple. You have a control sample and you run it on an LC and a mass spec, and you get, in this case, about 800 different peaks. Now, obviously, when we look at that, we don't see 800, but the output from the instrument says, well, there are 800 little peaks in there. Okay, so that's the control. You run that. The next run you make is a treated sample, so there's very little variability between analytical measurements. So this is a treated or disease sample, and you can see a big peak there, right there. And you want to identify that peak. And you know, I'm making this very simple. Just one peak. What is that thing? Out of these other 800, you just want to focus on that one peak. Okay, so with your instrument, and other speakers talked about the very good instrumentation that's available to measure exact mass. So let's just say that this has an exact mass of 268.1047. You know that very accurately with plus or minus 5 parts per million or whatever. And you want to know what that is. Pretty simple problem. So what we do typically is we go to a database. Everybody knows this probably here, most of the people who have talked. We go to a database and we ask which compound in the database has that exact mass. Okay. So the databases that have fewer numbers of compounds include these. They are very, very good databases. They have anywhere from 40,000 to maybe 100 or a couple 100,000 structures, chemical structures in their database. These include the human metabolome or metabolite database, Metlin, KEGG, these databases. So these are the databases that are typically searched initially with this mass. Okay, these databases, as most of you know, they contain compounds that are known. So if you're doing an untargeted analysis, and if your compound is not in one of these databases, you're not going to find it. Or you maybe find that mass, but it's not the right compound. When you look a little closer, maybe you buy a standard, it's not the right one. Okay, so these databases only contain compounds that are known to exist. And these databases typically only include about 20 to 30% of the peaks that are detected. Most of the peaks that I showed you in that chromatogram would not be found. You know, about 70% would not be found in there by exact mass. Okay, so what other databases can we use? There are much larger databases out there. For example, PubChem. PubChem now has, I think, about 90 million structures in there to search. So if we search PubChem with that exact mass, and if we even make it simpler and say that we're going to use an exact elemental formula, which I have down there, C10H13N504, which has that exact mass, and we're going to assume that is the right elemental formula and not worry about the accuracy of the instrument using that mass, use the exact mass because we know the formula, and we search PubChem with that exact formula, So here's PubChem. You put in the molecular formula. I've got that right here. You search PubChem, and you come up with, as many of you probably know, a lot of hits. In this case, this is actually a fairly small number. You'll come up with thousands of hits. In this case, 1,060. We've done this many times. Sometimes you get 25,000 hits. Okay? So you will get a lot of structures, a lot of candidates that are correct. They have the exact right elemental formula and the right mass. And this is adenosine. Maybe that's right. Maybe it isn't. There are thousands of these. So, so this problem is not trivial. The small databases, the very well-curated databases, often don't have the compound you're looking for. The larger databases have just a lot of false positives, a lot of nonsense. Okay, so most unknowns are not in these small databases. So obviously, and the larger databases have a lot of false positives. So monoisotopic molecular weight, even elemental formula, just isn't specific enough. Almost everyone in here knows this. Now, we also can collect collision-induced dissociation spectrum from our unknown, but those data are not in PubChem, and they're not included for a lot of the compounds in HMDB or Metlin. Some of them are, but not very many. Okay, so the CID spectrum that you get doesn't help you a lot in most cases. Okay, so what can we do? How can we maybe solve this structure identification problem? Okay, if we go to PubChem, there's some chemical and physical properties that are listed in PubChem. This is an example here. So these are computed properties. Molecular weight is obviously one that PubChem has for every entry. Log P, most of you know what log P is also included for all 90 million compounds in PubChem. Now we know that log P was not measured for all these 90 million compounds, right? You just can't do that. Okay, so how do they come up with this value, the log P value for 90 million compounds? Most of us know how they do that. They have a model. People have measured the log P values for about 30,000 chemical structures, and they've developed models then that will predict the log P for any structure. And that's what they do. So they use these models, which are pretty good. We know they're not perfect, but they're pretty good for predicting log P, and that's how log P value is included in PubChem for 90 million different structures. Okay, so this is sort of the idea that we're using in my lab, is to develop computational models to predict descriptors like log P so that we can predict those features for unknowns. So we could predict, for example, a CID spectrum for all 90 million compounds in PubChem, or a retention time for all 90 million structures in PubChem, so that we can search PubChem or large databases by filtering all the candidates. Okay, so using our mass spectrometer and our HPLC, we can measure these descriptors, or some of these we know. We can measure exact mass. We can measure a retention time. We can measure something called an Econ 50, which I'll describe in a minute. We can measure, because we have a Synapse G2 now, we can measure drift time on an unknown. We know that it's biochemical if we collected it from urine or blood or something like that. So we already know that it's a biochemical compound. And we can also measure a collision-induced dissociation spectrum for that unknown. So anything that we can detect in our sample, we can measure these or know these characteristics, these features. Okay. So can we build models, computational models, in other words, to predict these features for any unknown in PubChem, for any structure in PubChem? Exact mass, that's already done in PubChem. Retention time. Can we build a model that can predict a retention time for all 90 million compounds? Econ 50, can we build a model that will predict a drift time based on a structure. Can we build a model that will tell us whether or not a structure is a biochemical or a synthetic compound? And can we build a model or use models that will predict a CID spectrum for all 90 million structures in PubChem? So that's what we're doing in the lab now, is we're building these models by measuring, using knowns to measure these features and build computational models. Okay, so let's start with retention time. What we have done now is we've measured the retention time on 2,000 known structures, and we built an artificial neural network model to predict retention time. It's actually retention index instead of a model. So we use index molecules instead of time, because an index set of molecules can be transferred from lab to lab. So we have a group of alkanes that will standardize retention time, a C1, C2, C3, C4, and you run these with your sample, and then you can predict a carbon number instead of a time. This is an earlier model where we used only 495 structures, but as I said, we're up to 2,000 now. And the model works fairly well at predicting retention time for a given structure. It's not perfect, but it works pretty well. Okay, Econ 50. This is the energy, and it's center of mass, because it depends on the collision gas. If someone's using nitrogen or argon, that influences the energy that's required to fragment a molecule. But this is the energy required to fragment 50% of a precursor. And it's independent of collision gas because we standardize it using the model. Okay, so an unknown, a precursor, goes into the collision cell. It collides with collision gas, these little yellow molecules here, and starts to fragment. With initially low collision energy, it doesn't fragment. As you increase collision energy, this precursor will start to fragment, breaks up into pieces, and the precursor disappears and the fragments start to appear. And so what you get when you do the analysis is something like this. You initially start at a low collision energy where there's no fragmentation. The precursor represents 100% of the signal. You increase the collision energy in the collision cell. The precursor starts to disappear, and it goes down to 0 as you fragment all of the precursor. The value right here is called the energy in electron volts required to fragment 50% of that precursor. Okay? So you're just looking at the precursor, basically. Okay, so this gives you an idea of what we are hoping will happen when we use this descriptor, is that it's a good indicator of the overall structure of the molecule. So I've got some structural isomers, 4 structural isomers here, all having exactly the same mass. We're just moving this group around the ring, or we're changing that functional group a little bit. And these are the Econ 50s and the standard deviation of these isomers. So this is 15.6 electron volts. This is 16.5. And you can see the standard deviation is pretty small in each one of these. And so just moving this from para to meta gives us about a 1 unit electron volt difference, and it's statistically different. So we're in the process of building a model that will predict Econ 50 for unknown structures. Right now, our model has a very low number of compounds, but works pretty well. But we're expanding on this now so that we're able to predict Econ 50 values for any unknown structure. Drift time is another feature that we can measure with our SYNAPT G2. The idea is that a compound will drift through an inert gas depending not only on its mass, but also on its shape. Okay, so I've got 4 different shapes here, all with exactly the same mass, supposedly. And you might predict that a compound with this spherical structure would drift faster through this mobility cell compared to something that has the same mass but a more elongated shape. And so you can separate things with exactly the same mass based not on mass but on their shape. Okay, so we built a model with a small number of compounds to try and model this, and the model, even with a small number of compounds, works pretty well. This descriptor is not entirely dependent on mass, as you might expect. But as we get out to the higher mass ranges or higher drift times, there is some variability. So this may be useful for compounds, say, above 500 Daltons or so, where shape becomes more of a factor. The smaller compounds, it's probably not that critical because mass will determine drift more than shape. Okay, remove biochemical compounds. You might imagine if we were to search PubChem that has 90 million compounds, a lot of those are not actual biochemical structures. In fact, maybe most of them. In fact, most of them are not. Okay, so there are a lot of false positives in PubChem because they're just not biochemical structures. They're not good candidates. So how do we try to get rid of these false positives that aren't biochemical? They're synthetic. The idea is pretty simple. If I give you guys, any of you that are analytical chemists, a structure, you can instantly tell whether something is biological or not, probably, right? It's got triple bonds. It's probably not biological, and other things. So it's similar with skeletal structures. We recognize all of these as biological, and we can actually recognize some of them as being related. Primates, fish. This is an elephant, obviously, from the trunk. A dog. But if I were to give you this structure, you would all instantly say, nope, that's not biological, right? I just sense that. So a lot of us, after looking at a lot of chemical structures, can look at a structure and instantly say, that's not a biochemical compound. The question is, can we teach a computer to do that? Can we teach a computer to look at a structure and say, yes or no, it's biochemical? The idea is pretty simple, just like the skeleton idea. Metabolites have evolved from simple precursors. So they're all the metabolites that we have are all sort of related in a way, just like our skeletons are. So if we ask whether a structure has these types of components or not, in other words, if we take biological scaffolds and ask whether these are in a candidate compound, if we see a lot of biochemical scaffolds present in a compound, that indicates that it's more likely that this is biochemical. If we see things that are not biochemical, like, for example, things like this, if we see these in a chemical candidate or structure, we sort of know that it's not a biochemical compound. It should be thrown out. So this is software that we've written about 4 years ago. It's called Biostructure Matcher. It determines whether something where the chemical structure is or is not likely to be a biochemical compound, and it kicks it out if it's not a biochemical compound. And when we use this on a representative sample from PubChem, we see that about 10% of PubChem is likely to be biochemical. The other stuff is not likely to be biochemical, and it can be removed as a likely candidate. So that's a pretty good way to discriminate among compounds. Okay, CID spectrum is the last thing that we can measure with our instrumentation. It's a really, really unique feature for every structure. The problem is the theoretical basis for CID fragmentation is very, very poorly understood. It's very, very difficult to look at a structure and predict what the CID spectrum would be. Okay, so this is a problem that we're working on. It's not easily solved. Again, the idea is you take your unknown, you put it in the collision cell, you fragment it into the various pieces, and you look at the mass of the pieces as well as the precursor. And this is the experimental CID spectrum. The idea is if you have some compounds from PubChem or HMDB, some candidates, you look at their predicted CID spectrum, right? You have their predicted spectrum that the software might predict, and you compare that with the experimental CID spectrum, and the one that matches the best is more likely to be your correct compound. Now, there are a lot of problems with this. These CID spectra, these experimental CID spectrum, a lot of people assume that the height of these peaks represents the abundance, but it really doesn't. It depends on your experimental conditions. It depends on your collision gas. It depends on your collision energy because they're scattering. If you have a high collision energy required to produce this spectrum, you're going to lose a lot of signal from the small fragments because they fragment and disappear. I'm sorry, they scatter and disappear, and you're not even going to see them. It depends on the RF setting in your collision cell. That really strongly influences what your CID spectrum looks like. So there are a lot of problems with doing this, but still it's a very, very good feature for trying to identify an unknown structure. If you can exactly match a CID spectrum, you've got a good idea that you're unknown. Now, this is very time consuming, and so trying to fill databases with experimental CID spectrum is just not going to work. So hopefully there'll be some advances in this area of trying to predict the CID spectrum, but that's a long way in the future, I think. So the overall idea with this approach is for any unknown that you want to identify, measure its mass, retention time, an econ 50, a drift time, a CID spectrum, then go to a database like PubChem, something that's maybe likely to have the unknown in it. You filter these. For us, our chemical space only includes 6 elements: carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. We're not looking for anything with a chlorine. So if we're going to PubChem and it's got a chlorine, we kick it out. Bromine, out. Fluorine, just these 6 elements. We remove anything that's got a non-biological structure, double bonds like this, triple, cyclic propane type rings, kick those out. And then we get, with this mass filter, we get a group of candidates. The example I showed you before had about 1,000 candidates when we used that elemental formula. So let's just say we have 1,000 candidates that have the right mass and are biological and only have 6 elements. Then we ask which of these 1,000 structures have the right predicted retention time. We run each of these through our model. The model will predict a retention time. We compare that with our experimental retention time, and we kick out the ones that don't have the right retention time. What's left, we predict an econ 50. We compare that with our experimental econ 50. Those that aren't in the window, we throw out. We do that with drift time. We do that with, then we try to predict or rank by CID matching. And hopefully at the end, we have a very, very small list of candidates, maybe 10. Often it's not that small. It's usually never 1. And hopefully our list contains the unknown, and we would then buy or synthesize that compound and compare it with the actual unknown. And maybe it's right. Maybe it's not. Okay, so hopefully if this works, if this strategy works, I'm not sure it will or not. We're not far enough along. But if it does, the idea is like log P in PubChem, what we would like to see is we would like to see some additional computed properties. In addition to exact mass and log P, we would like to see PubChem list retention time, actually retention index, for all 90 million compounds using our model. We would like to see PubChem predict an econ 50 value for all 90 million compounds. This is just an estimate or an example here. For all 90 million compounds, a drift time. We would like to have PubChem tell you whether or not their input is biological or not, and for PubChem to have a CID spectrum, the computational CID spectrum for all 90 million compounds. So when we measure something, an unknown, we measure all of these. We can just go to PubChem and search, just like when you go to Google and you put in 5 descriptors and you come back with 1 hit. We'd like to be able to do that with 5 or 6 descriptors by searching PubChem. Of course, that assumes that the unknown is in PubChem, which, of course, is not necessarily true. So PubChem doesn't have all these unknowns. So that's another problem that exists, is how do we build better databases to include all potential unknowns? And as you know, chemical space is huge. That's going to be a big problem to solve. We're also working on trying to improve these models. As I said, some of the models have very small numbers of compounds, so the models aren't so reliable. But that's where we're working with the model prediction. So now, so how can we identify unknown unknowns? That's another problem, and we're working on that as well. I don't know what happened there. I wanted to show you a list of the people or pictures of the people who are helping with this. In the lab, Dennis Hill works with me. He's been working at University of Connecticut for a long time. A clinical investigator. We do trauma work in children. He's at Children's Hospital up there. Some postdoc and some undergrad students, and a person who helps us with the modeling at Massachusetts. And I will stop there. That's my last slide. So if you have any questions. You're welcome. Thanks.
I think we might have time just for one quick question. Is any?
Yeah. Either one.
That was a spectacular talk. Thank you very much for it. So, what does it take to do your prediction on those million compounds? Is it a logistical hurdle, like more resources to essentially measure all these more than the 100 or so that you listed on your predictions, or what will
It's
it
two
take?
things. For example, our retention time model that we have now, it's fast, it works really well, but it's not accurate in certain retention time areas. For example, compounds like lipids that have longer retention times, our models don't predict very well. So we need to add another 1,000 compounds in the high retention time area to better predict that region. So we just, we kick things out that shouldn't be kicked out, basically, because we can't predict them well, accurately.
So money and a human resource.
Yeah. Did you have a? Okay. Okay.
Hi there, Nick Ratcliffe from Yale. I just had a quick, more technical question about your eye mobility modelling, and
Yep.
you took drift time into the equation. Did you take collisional cross-section of,
Yes.
of the molecules as well? Did you do any in silico collisional cross-sectional work as well?
Yeah, there's a formula that does that that we used. So that was incorporated into the model for most of those compounds. Yes, we calculated their cross-sectional areas as well as measuring some of them.
And I know there's a group in Bangor in North Wales, believe it or not, that have actually got a really big library of in silico collisional cross-sectional values.
Are they pretty accurate?
I think
Yeah.
so. I think they're
Yeah.
done on the exact same. I think they're done on the exact same instrument you've got. So.
We used the one that was developed by the guy at Purdue, Dustin. You know his Clemmer, yeah. Indiana, okay. Yep.
I just have a quick comment. Are you aware of I bet you are. Canada has invested about $20 million on characterizing the metabolome from tissues and fluids. And those
With
data,
David Wishart?
Yes.
Yeah.
And those data right now are publicly available for people to maybe, I don't know, have you considered incorporating this data to your system?
Now, what did you say they've included?
Metabolome.
Yeah.
Like the serum metabolome. They have
So
used,
they're just adding additional compounds to their
Yes.
40,000 or so now?
Yeah, yeah.
Yeah, that's great. What's your question, though?
Your question, my question is there any way that you can, based on your system, that you can incorporate this kind of libraries into your training, your algorithm, how to identify?
Well, to build a model, we buy about 2,000 compounds or so. You can't buy these biological compounds. They're too expensive.
No, I know. What
So-
I'm saying is, can you use the data from them to incorporate in your model?
Probably not, because they probably didn't measure retention time. Or if they did a different system. They used a different system. We're using reverse phase, heptafluorobutyric acid. So even though our models try to be as general as possible, it still depends on how you measure these compounds. So for our retention time model to be of use for anyone, they would have to use reverse phase heptafluorobutyric acid and a C18 column. So if those data are available on those compounds, then perhaps we could use them, yeah. But otherwise, no. Yeah.
I just want to ask something about your retention time, but you answered the comment. your retention time is just based on this LC method, because there are so many methods.
Right.
And how are we going to deal with this problem? Because it would be nice to have all those predicted data, but retention time.
Right. It requires that to use our model, you'd have to use a C18 reverse phase column with heptafluorobutyric acid. So it's ion pairing. It's not too much to ask, I guess, but it is required, yeah, to use the model. A lot of people use reverse phase, so yeah.
Yes, it's everyone join us.
Oh, thanks. Appreciate it. The problem is reviewers want the answer in 4 years. It's not going to happen. No, I have not. Yeah. Yeah, I didn't even thought about that. But it's going to take a lot of work. I mean, you know, measuring 2,000 compounds. Yeah. And a lot of people don't want to standardize their conditions either, which I understand.
So
You know,
hi
you
everyone,
don't
I'd like to
use
thank you
reverse
for joining
The other
us
thing, we're
today
just
and
looking
also
at
for attending the roundtable discussion. I think we can all agree that today's speakers have really given a wonderful overview of the recent developments and applications of the exposome, and also some of the challenges that have arisen as well. I'd like to reintroduce some of the members of the panel for those of you who may have joined us later. So we have on the end Dr. Chirag Patel from Harvard Medical School, Dr. David Grant from University of Connecticut, Dr. Toby Athersuch from Imperial College London, and Dr. Siraj Dangana from Waters Corporation. If anybody has any directed questions for the panel, we'd really like to hear those.
So I just wanted to follow up with a question to Professor David Grant. You mentioned you'd like to see all that information put into PubMed, and one of the challenges that kind of was hinted at, but I think it's even more than the one that was mentioned, was retention time. I think it's going to be very specific on the instrumentation, column, mobile phases, solvents, percentages, but I even think EC50 is going to be instrument dependent. The collision gas is going to be instrument dependent. And the other parameters, which were, I'm trying to remember.
You're right. You're absolutely right.
There was one other that's going to be extremely instrument dependent. So I don't know how.
CID spectrum.
CID, that's the one. Thank you. Instrument dependent. It would have to be on a specific instrument with a specific column, almost plumbed identically, and controlling those guesses. I am interested in how you think PubMed would be able to do this and keep all those things controlled.
Right. It's not like we're doing log p-values, which octanol and water, and which is pretty simple. So I don't know. I don't know whether people would adopt to something like this.
So my follow-up question is to move from what I would call more a meandering way to a higher throughput way is, as you start to identify things, can you build an in-house database of things you have identified so that if you run across it again, you don't have to go through the same process?
Yeah, I think that's the strategy now, is when anyone does a metabolomic study, they send their data to a repository, and that's, I think, funded currently. And so all that data is available. The problem is a lot of the information that should be included, like the collision gas, the RF voltage, things like that, really aren't included in a lot of cases. And so even the data that are put in there, you can't really replicate because you're not sure what parameters were used often.
But your lab could, because you will have the EC50.
But
that,
we can just run a few compounds. I mean, we
right,
can
but as you do it, you can build it internally for your lab.
the models that we use, though, we're using compounds that we can buy that cost about $10. They're not metabolites.
Okay.
They're screening compounds. And so we'll take a C8 screening compound, and we'll add a nitrogen group, and we'll buy compounds that move that nitrogen group down the chain so that the computer model knows what to do when it sees that. So it's a very systematic way that we buy the chemicals and the chemicals that we get so that we inform the computer how to deal with these different structures. So they're not biological chemicals that we're using, actually, to develop the model. So they really wouldn't be that much use, these 2,000 that we have in there, for example.
But if you did an unknown and it wasn't
Yeah.
metabolite, you could start to populate.
We could, yeah.
Yeah, I would like to add, to your question earlier that yes, there are ways we can start. If you are generating a data set and you have a fragmentation spectra that belongs to certain compounds, there are ways of starting to populate your in-house library, for your own, increasing the throughput of your own workflow. It might not be very, universally compatible with other systems, but internally, yes, there are ways that can be done. Yeah, but and also I think we're starting to realize how we can model. For example, collision cross section is one area. Different vendors calculate that differently, but it's for the most part a gas phase measurement, and with some adjustments, we can really translate one set of measurements to another as well to make them usable for other platforms as well.
Yeah, anytime you can have a set of standards that you run with your unknown, like retention index or drift time, the phenylalanines that you include with your sample. So it's indexed to known compounds in the sample. That really helps.
Yeah, from a toxicology metabolomics perspective, we know that something like 8-hydroxyguanosine in urine is an indicator of oxidative stress to DNA, and this is jettisoned as it's repaired and ends up in urine. So who knows? There may be a lot of damaged DNA bases running around in blood or urine that nobody's looking for. I'm just wondering if there's much literature on looking for modifications to DNA, whether it's on an epigenomic basis or on a repair nucleotide basis that people are starting to look for.
Well, it's not my particular area of expertise, but historically, a lot of people have done what was basically the old, if you like, adductomics. That's how we get those kinds of measurements. I think in terms of what's being described here, that isn't one of the focus for foci that I've seen in analysis. I think people are looking more at other classes, bile acids, lipids, things that dominate particular matrices, I think, at the moment. Although I take your point that I think as those classes are well served by particular targeted analysis, probably best that. But I'm not particularly aware about. I don't know if anybody else is. No, sorry, just yawning. Okay.
Well, and the other, that's oxidative. It's an indicator of oxidative stress to DNA, 8-hydroxyguanine.
Sure.
Another would be oxidized lipids, diene conjugates, or other biochemicals that we can measure in blood or urine as a quick screen of a population, rather than having to get a liver sample or something in lymphocytes that may be indicative. I mean, is there something easy to measure that's an oxidative stress marker in a population, off of lipid, off of DNA, off of protein?
I see what you're saying now. Okay, so related more to the oxidative stress. I think oxidized lipids, definitely a good place to go with that. That said, I think that there's probably already the markers we do have. If people are interested in that specific question, using sort of targeted approaches where you've got there, I think if those classic markers we don't already know about and we couldn't just measure, I think that the untargeted approaches, particularly for lipids, will definitely, it will capture a lot of that, those metabolites there. But I guess it's just, again, I say that's not my specific area of expertise, but I think that it would be well served there. I don't know if anybody else has any comments on this. No? Okay.
Well, it would be interesting to know what endogenous levels are of those kinds of things over time in humans, just to get, background information. That would be good, yeah.
So I think one of the other things about the lipid analysis particularly is just the complexity of the lipidomics profile. So if you look at the lipid maps database, they go bananas on the complexity of lipid metabolism. So the thing I was describing earlier about there being, in some cases, people having different, parallel and complementary analytical platforms. That's one way that can be addressed. We're having a specific lipidomics assay that's run in association with others. For many, I think that people doing a sort of one-shot reversed phase analysis, it's just not got enough resolution to be able to go in with such exquisite detail to look at across all of those things that you might imagine that way. But so I think the way I would go forward is if you can isolate, if you like, or if you want to partition out lipid analysis in that example, that would be good. I think the sort of traditional approaches for the DNA adducts,
then you have to get DNA and isolate that, and if there's something easy in urine or in blood to sample, like 8-hydroxyguanosine is used now, but there may be many other markers that might be sensitive to different types of stresses and compounds.
And one of the things that I'm aware of that lipid has been used, especially oxidized kind of lipid or lipid profile, is to assess the quality of the sample. So because, we may not know the history of the sample, how many freeze-thaw cycles they have gone through, and how much is exposed, and lipids inherently oxidize. So those oxidized lipids have been looked at, some of them to assess the quality of the sample, and perhaps, it might confound us, but might be the same approach we can use to assess some of this oxidative stress as well.
A lot of lipids that they may alter, but the tricky part is to see which lipid links to your biology, because a lot of them might be oxidized, may be altered, like concentration-wise. You can find all types, and a lot of families, groups of lipids can alter, but then you have to go back and have to have you have to find the pathway. How is this linked to your biology, and then go back and see, okay, it's a good biomarker. So I think this is also a really challenging point. Have to think about it.
I've got a really different question, and it's more about the future and where the future lies. So, from the talks that we've seen today, I think there's sort of 2 or 3 main sort of things that will push exposome analysis sort of to the levels that we want to get to be. And I think one of them are technological advancements. Maybe 40 years ago, a single quad mass spec was the size of that lectern. Right now, I know that Waters produced one that's probably the size of the PC that's running the projector. And so the other aspect to it is sample size, is being able to get up to the levels of, or metabolomics needs to get up to the levels of GWAS studies, millions of samples, tens of millions of samples, even billions of samples. How are we going to do that? And I think one of the main things is probably this guy here, is the mobile phone. And what do you guys think about the technology that's going to happen in the future to be able to allow something like this to be able to pick up all the chemicals that we need to be able to see, and then maybe the data basically what data processors are going to have to be able to handle all of that data. Because I think that in 10 years, 100 years' time, that's probably what we're going to be facing, probably what we're going to be looking at.
Well, I've always said it would be fantastic if we could put an NMR inside a mass spectrometer. You know, and I know that's not easy, but we do need something, it seems like, a way to combine NMR and mass spec in a better way, so that NMR can be used on low abundance metabolites, not, you know, just the 44 samples or so that the NMR can, because it's so powerful and so quantitative. Some other technology that may be useful in the future, I'm not sure, but we're sort of looking at, is the use of lasers, high energy lasers to fragment precursors. That may be useful as a better way to get a CID spectrum that's independent of collision gas, scattering, things like that. So just some ideas that I had.
That's a great question, and I do also share your belief here in anticipation of this stuff being more omnipresent and cheap, essentially. I think, but in the near term, what will be immensely useful, and I think is proving useful currently, are biobanks. So, you know, samples are connected to clinical phenotype information that we can measure, you know, things like blood, that we could do the correlations just out of the box without having to deploy those devices. So I think, you know, for example, UK Biobank is there to do measurements with and accessible. There's a Kadoorie Biobank in China collecting on the order of 500,000 samples. Existing epidemiological cohorts have the biobank. So I think, you know, this is also a huge area to exploit as well.
So I remember several years ago that there was an article came out relating to a paper that had been published related to sort of metabonomics, and the idea was there was a tricorder. It was the whole Star Trek thing, right? And you know, like 10, 15 years later, and we still have the same vision that would be a really cool thing to do, and it would be. I think in terms of the platforms we're trying to do, obviously mass spec has potential to be minimised. I think it would be limited with NMR platforms for the near future at least, just due to just the operational our ability to do anything useful with it. Although low field benchtop NMR might prove something useful if you were able to build models with relatively low resolution data, but we're not down to the same sort of size of doing it on your handheld. But other platforms that people do use, like IR and Raman spectroscopy and things like that, I wouldn't count them out as being completely unuseful, because the thing you're talking about there is potentially that we, in one hand, we trade off the resolution and that we get from these very high resolution instruments and ability to look in good detail at all of these samples, for something where it's good enough, but if you've got a billion people who, you know, if a billion people just took a picture of something we asked them to take a picture of, even if it wasn't a very good picture, we'd have enough pictures of it to say something useful. I don't know what it would be. I think there was an example of everybody took pictures of the stars during there's like a astronomy program in the UK, and then everybody took pictures of the stars with their cell phone, and then they're like, oh, we just discovered a new galaxy or something. I don't know. It was something like that. The point was, it was crowdsourced. You couldn't have got a telescope that big from all over the UK, all doing it all at once, and just all for free anyway. So I think that obviously you have to sacrifice a lot because you can't get all of the things you want in the phone. But something there, maybe a sort of halfway house would be if we were able to push for sort of citizen science type of projects, where maybe even if you don't have it in your phone, but there's it becomes sort of a commonplace type of thing as you do, you know, your general things to do with the health service or whatever. You submit samples to a common core, or there's just a very simple machine that you just put stuff into. I don't know. It's a knotty problem because with that it comes with the fact that all of those people will have to operate the thing the right way and not abuse the system and all of these nightmares that we might have with that. But I imagine the fact that these are being addressed in the personal sensing arena, it won't be long until we have some sort of capacity to do spectroscopy, I guess, just out of sort of thing. Yeah. Yeah, that's pretty good. Isn't it? Yeah. Yeah. Well, I don't want to be like that person who was quoted earlier saying that will never happen ever. So I'm not going to say that. How long will Waters take to develop a handheld multi-spectrometer?
Well, maybe there's already in works. We don't know. But I think, going back to your point, I think we can, with the technology, and we saw in the course of the talks today, in mass spectrometry itself, we have advanced quite a lot. We're really pushing the limits. And I think when we talk about exposomics, especially looking at things in the unknown, we're always driven by what can we find more, at what low level. I think that's the direction we're going to see. And also by using how little of a sample. So can that then take us into the realm of like chip-based technologies, right? I think that's where we might be headed. But one thing we do have to keep in mind is every time we're trying to do this large cohort studies, reproducibility is key. We might be able to push the limits with sensitivity, chip-based technology, nanoflows, but can we have that measured reproducibly over the course of a large sample set? I think a lot of it is going to be driven by that.
Let me have one point. I just want to tell, everything was great today, but I'm always, just like Gary, would like to see some biological effects trying to translate this data. We didn't touch today at all the tissue imaging mass spectrometry, which I think is also the future in this, which we're developing here, and Waters is giving us a Synapg2 SI with a MALDI and ion mobility. And we're also organizing next November, we're going to have another symposium just like that on tissue imaging using mass spectrometry. But what I wanted to tell you is one of the things that we're facing, this was mostly for Chirag, is this, and it was mentioned throughout this today, how we can have these two the omics talking to each other in terms of big data. How you can get this, and we just we got funded from the Eye Institute to do the eye tissue imaging. We have some preliminary spectacular data already with Vanderbilt, Caprioli and Kevin Sai. And we're moving now, we're developing it here. But one of the things that we also notice is how we can get the pathways to talk to each other. So we put an application R21 to the Eye Institute, but they want us to do more programming language-wise. So how do you think that this can be solved in terms of not only simple pathways? I mean, there is no really a good software right now that you can put your transcriptomic data, you can put your metabolomic and lipidomic data, and then try to see. I mean, Agilent did try some on a software, but you have to loosen up your stringency and everything, and you just have to do a lot of things which can go back to your bias thing that we talked about. So what's your opinion?
Sure. So in my work, I've noticed that the one of the first challenges that we face is having all the data in one place. Okay, a very simple logistical issue is just having the genotypes, gene expression, metabolomics, and the other phenotypes all in one place. Okay, so I think leveraging existing resources that have already done these measurements, the genotyping, the expression, and having the tissue specific expression specifically, is one way to address your need. So for example, there's the genotype tissue expression data source that you may have heard of. It's called GTEx for short. I think this could be one repository where you could access those samples, do your phenotypic measurements on, and have the genotypes and expression measurements essentially for free, because it's all there. I think it starts with resources like that to build a database that have everything in one place.
But my point is, how you can have those crosstalk with each other? That's the trick.
Yeah, that's a trick, and I think to appeal to other data analysts, I think it takes large sample sizes, and but I do think that the methods are becoming quickly commoditized. Like we can really download our packages to do a lot of these things. But in my experience, is having everything in one place. That's the limiting factor.
Crystal, can you hear me? Crystal Paula, UMass Amherst. Thank you for your talks. So Toby raised an interesting point of citizen science, and with like the life study and then all the Helix projects. And this is going on a different tangent, but do you then try to engage with the populations that you're working with and then attempt to communicate those exposure back to that population in terms of an intervention or even engaging on what their environmental exposures are?
So do you mean directly to the participants about the data from that study, or do you mean more generally do you feed those data back? Because, for example, for the Helix study in Exposomics, the whole purpose of it is to help improve Burden's disease, modelling of disease risk, and these kinds of things, are to make policy changes that influence, well, improve people's lives, right? But in terms of communicating information back about the study, there are, for most of these, most studies, whether they're large scale or not, mandated communication packages within there that to distill down the main findings of those studies. On an individual level, I think that's down to the sort of a combination of the ethical review of the project, what's being measured, how that's considered, what can or should be communicated with individuals. Obviously, if it's miscommunicated, that can be probably a bit of a nightmare for everybody involved, because well, for obvious reasons. But I think that there's a general will to be engaged with the people who are there. I mean, if you think about it, all the people who are researching here, I was saying earlier about the people who have developed these biobanks many years ago, also the people in those biobanks, or not people in them, but who've contributed to those biobanks, that's what we need. We rely on the goodwill of people. So I think it's actually maybe not necessarily just the communication of what their results are, but more general communication about the fact that these are the things that are able to help you, well, collectively, improve your life, improve the community that you live in, and more generally. So I think one thing that could be probably communicated more succinctly is, or more effectively, is that being involved in these kinds of projects is, what I would consider a sort of, not a duty, maybe that's a bit of a too strong a word, but it's something that you should seriously consider would be just a normal thing to be part of. I think those people who do enroll in these studies, then they may feel that way, but it's not. I guess that's not always the case. Did that make any sense? My point was normalise being part of doing science is my point.
So I completely agree with the point you're trying to make. I think without engagement, we're not going to get, from a researcher's point of view, we're not going to get the samples that we need to get robust associations at the end of the day. So, we definitely need some sort of engagement. And I think the exposome itself, can provide an educational opportunity. I think of the genome, for example, it provided that type of opportunity for individuals to relate to. So now people think, not most, but many people think of the genome as a way to understand their ancestry, certain things about genetic risk for phenotypes. And I think if we empower individuals like that, and of course we can increase sample size, but there is this opportunity for understanding how the environment in concert might, affect our, people's health. So take it for what it is. My short answer is no. I don't engage at it right now, but to achieve sample sizes, for me as a researcher, it's compulsory.
Well, you yourself found that exercise was very important for health. How would, I mean, how can we get people to exercise? I mean, even if they know it, right? Even if they know they should eat well and exercise, it's still a problem. Even if they know they shouldn't be exposed to, some chemicals or, avoid PCBs or, I don't know. It's just, it's a problem. Even if you have the information sometimes. Education is a real strong part of that, I think.
Hi, my name is Casey. I'm a student at Southern Connecticut State University, majoring in public health. And similar to that, my question is, do you work with the initiatives, like the global initiatives that you spoke of today? Do you present your research to them, and then do they implement programs to provide the education to specific populations? Like, where does your research overlap? Because you're saying that, how do we get people to exercise? Well, with those initiatives, are they the people facilitating and educating populations to be able to make that change? And are you aware of those changes?
Okay, so for the projects I've been involved with, there's not a specific remit necessarily for an educational program within the individuals enrolled in the study. There may be in other studies, I don't know. I think the main communication that we have is that we have a strong framework for explaining or describing what this whole exposome concept is, as an environmental complement to the genome. That's quite a clear concept. People can get behind that maybe. But the way to communicate that and to education is really for policymakers. For us, we need to present the best case science coming from the research packages that we have within these projects. So from my point, can I delineate which metabolic responses are most important? Can the epidemiologist decide which of those are most highly correlated or related to outcomes that people are interested in? We publish that results, and then we have sort of a translational group who help policymakers interact with those kinds of data. So it's not a case of me just going up to someone saying, hey, listen, you need to educate everybody about this. It has to, from the way I understand the way these work, we have a sort of, our responsibility is to present the science, and then those who facilitate sort of science or evidence-based policy making, who then initiate a set of strategies based on whatever other priorities they have to either do education or cause public health interventions or what have you. So I think I can't speak for everybody, but I was saying we just need to present the best case science we can, what we said we will deliver in these projects, that then will hopefully come forward collectively to deliver those policy changes. Does that answer your question?
So, being in a school of public health, that's a very important question that you're asking, is how does science inform the public, and what's the best way to do that? That's a very, there are a lot of people now who do not believe in global warming, for example. Why? Why don't they believe that? Obviously we can't prove it's happening, but all the evidence suggests that, and yet people ignore that. Why is that? Is it an educational problem from a very early age? Do you guys address that in this school? What I learned in school is a lot of intervention,
And currently when we're provided with scientific research, sometimes our goal is to think and create different ways how the public will retain it the best. And that's where additional research is sometimes needed, because who's our target audience? Who is retaining the information? Does it start at a young age? Does it start in college? And I think it is just a cyclical thing of questions, and where does, I understand what you mean, how does it just happen?
Where does critical thinking start?
Where does it, isn't it, I could sit here and wonder, but You did answer my question. Now I'm getting off task. I don't know why.
One other perspective on the public health and educational part, there can certainly be unintended consequences. So when we alert as a Department of Public Health the public about fish consumption, PCBs, mercury, we find people aren't going to go near fish at all. And that's not our objective. Right. You know, so to have a mixed message, a complicated message, it's almost better to not have the message at all because people are going to do the wrong thing. And I do have a question.
Vagelis Dioletis, and I'm a postdoc here at Yale. I would like to ask you if you think that we are going to define in the future a healthy exposome in an ever increasingly polluted planet. If it makes sense to have a steady point of reference to compare to, to look for different diseases, different symptoms.
Can you repeat your question?
Do you believe that in the future we are going to be able to define the healthy exposome, in an ever increasingly polluted planet? I mean, this is particularly important, for studies that, have a great deal of time in terms of duration, and especially when we have great variation in geographic pollution and new technology, new products. Seems to be a very dynamic feature. And yeah, do you think we're going to be able to outline or define the healthy exposome?
That is a tough question. I mean, maybe. On behalf of my group here, I think we all agree that's a tough question. I don't think we still are at large of, the question at large of what is good health is still, something is one cannot answer today, with or without the exposome. So I think it's a very tough question to answer. Albeit there are groups who are trying to do this, including Google. So you might have heard of Google Baseline. They're trying to ascertain all these omic measurements on on the order of 1,000 people, just to understand normal variation of these omic measurements in the population. There, of course, is NHANES, which is a non-institutionalized population of the United States, of which we've developed we, the scientific community, have developed how these clinical markers vary in the population. And so I think with approaches like this, just collecting information and to understand variation is the first step. I'm not quite sure how to get to what is health versus disease in general. But sorry to answer your question in a hand-wavy way. But.
Yeah, so it's a philosophical question, isn't it, there? I think we're considering the exposome as this totality of exposures over your entire life, from conception to death. That's what we've been talking about today. And I think that actually, obviously, a lot of the emphasis is on identifying which of those exposures have negative connotations or associated with subsequent disease outcomes, shall we say. But obviously, there's some fairly mandatory things you need to be exposed to, like anything to eat. You might need pharmaceutical interventions to keep you alive, surgery or whatever, and responses to infection. And I think actually, ultimately, it's a little bit of a moot point because you can't retrospectively change what's happened to you. And even if I could describe what a healthy exposome would be, I'm sure we could all come up with a sort of optimized, for one individual, what their optimized exposome would be. Due to the fact we're all experiencing this in a sort of gene versus environment thing. We all have different genetics anyway. So what's right for you is probably wrong for me. And so that's the context. So there's the genetic context, and there's also, for anybody who's already alive, there's also the context of all the things already happened to them. So we could, it's a nice thought experiment to do, but I think that it could be a bit of a time sink trying to define that. I think if you're sort of an E. coli, you could say, okay, I can define some growth conditions which optimize one particular bit of your life, you know, how fast you replicate or whatever. But that's fine. But the other thing is, if we're also defining those exposures, what might be optimal there, you might also, if you have the same question, you need to define what the outcome is you're trying to say what's it optimal for. I mean, if you want me to live for 150 years, but I'm really, really miserable the whole time because I've had to use your optimized exposome diet, right? That's not, you know, that's an outcome which not necessarily would be optimal. So, and again, you can't know that until you've done it, and because we're all different, you don't know. Anyway, so, but yeah, do you know? What is it? No suggestions? Okay.
Well, the other thing is, you really don't know what's the calculation, because you might select your sample, and maybe the people have some disease that you never know. So I think that makes things even more complicated compared to that.
It's probably a better idea to look at the resilience of groups of people rather than the actual exposome itself. So you can identify people that might be at risk of, as you said, poor environments, poor food, poor exercise, etc., etc. And I think in doing so, you will hopefully extend their life, because that's what a lot of the clinical interventions are for the end of the day, and extend the quality of life as well, which is, as you've sort of said, Toby, as well, it's vitally important. There's no point making people 120 years old when they're bedridden and
Well, there's the, I think the initiative, again, I'm not part of it, but it's like the 100, like 100 years of life, 100 years of health, described in whichever way you like. But that's a good point. And it goes back to this idea of, that I did mention a little bit, that I quite liked, is that as we are all individuals, don't lump us all in together, where you can define individuals' own sort of biological space, and they're happy there, or that might migrate through time. So I think the whole idea of this being a tailored thing, what's right for you is right for you. So in many ways, we're thinking very much like a sort of precision medicine thing, which is being developed, you know, in a sort of parallel track in a way, in a clinical environment, of saying, I know what you're like now, I know that I want to avoid you having these outcomes, and this would be a preferable outcome. Which of these interventions or no intervention can I take? So yeah, I think that's probably that's the key there. I think that was the heart of the question, wasn't it? It wasn't like, you know, give me a list of what you should or shouldn't be exposed to. It's like, what is a reasonable expectation of what you can identify for an individual or group of individuals that would be most appropriate for them to have a happy time. Okay.
Hi, Nicole Diesel, Yale. I want to thank all the speakers. It's been really fascinating to see this theoretical concept of the exposome actually being implemented, and I'm really excited to see where your findings take you. One thing that stuck with me was in Dr. Patel's presentation, you put all these hundreds of exposures and other variables into your models, and still your R squared is only 1%. So any suite of exposures will only explain a small percentage of the variability in a health outcome. And I just wonder how you kind of think about it, how you interpret it, especially as scientists trying to argue the importance of studying the environment, the importance of the public health impact. So I wonder if you could just offer your thoughts on that.
Yeah, it's a great question. Man, this is a great question. So 1%, if we look back to that heritability and trying to estimate phenotypic variation, 1% is not really that much, you know, as your question is alluding to. I just think it points to, we have a lot of discoveries to be made, okay? And so that's essentially one inspirational thing for, you know, getting funding to do discovery. But it also could mean that a lot of variation may be just due to, you know, randomness, right? And I think, you know, I'm trying to understand how to decipher, you know, one versus the other, and I think that is in fact an opportunity. But I think your real question is, like, how do you communicate what these low effect sizes to people that say, physical activity is not important. It's only like 1% of R squared in mortality. I think it matters in terms of how to describe biological, causation at the end of the day. 1% is not going to describe clinical risk, but that's a different argument than understanding biology behind, phenotypic variation. So I think there's this divide, and I think that doing this type of work while we only get 1% will be useful in helping us understand biology. Just like GWAS doesn't explain heritability all that well, but the variants that have emerged have been able to identify the risk for risk pathways, genetic risk pathways for disease.
Thanks. Yeah, maybe this is kind of the attributable risk type of question, and it's maybe something to think about as we try to interpret these findings for the public to present the results in a way that does kind of convey the potential public health impact. Thanks.
Yes, Guy. No, actually, I want to close, but... No, wait, I have one more question for Waters.
Oh, okay. So, as a toxicologist, risk assessor, we are always throwing around uncertainty factors, and the standard default one to try to get to the sort of safe level of exposure called the RFD that everybody could be exposed to, and we could all sleep well at night, not worrying that we're setting the number too permissive, so to speak, is tenfold between people. So from your experience of looking across these exposome and with some indication in some cases of biological effect, do you think that variability between people in terms of susceptibility or exposure is really just, tenfold, or in certain cases, especially from your work, Chirag, some of the things you're looking at, should we be looking at bigger uncertainty factors for some disease states and risk for disease? How variable are we?
So to answer your question, we're extremely variable, right? So if you take the exposures that in the same household, for example, one person doesn't predict the other person, right? And so, so I don't know. I think I have to punt on your question. I'm not quite sure how this translates to what that might mean in an uncertainty factor, but I do think that this has implications on who exactly is your target population. Perhaps it's perhaps you need to sample more people that live in a fixed environment to understand better that uncertainty factor, for example, rather than specific individuals. Like in a household setting, you'd probably want the entire household to capture the variation within that household. So I'm not quite sure how to answer your question, especially given the heterogeneous nature of a lot of these exposures as well. So maybe something offline.
We can continue over wine and cheese. We have a great selection. Before you start going out, I would like to ask Waters, Dustin, and Suraj, what's the future plan? How Waters is trying to accommodate the challenges of the future in terms of more higher resolution instruments? Is there anything? I don't want you to reveal secrets of your company, but are you planning to get some more advanced, high-resolution type of machines that Dean Jones was talking about? And how about, more advanced software?
Well, I think there is, I think, exposomics is such a complex area. We have to look at it in a holistic way. And then, like David pointed out and Dean pointed out, yes, resolution can get us only so far. When, like the example with, metabolite ID or a feature ID based on accurate mass, we can have 1 million resolution, but still going to get us to the same place as your example. So what we really need to do is take it at a holistic approach. We have to use all the tools we have in our toolkit, which involves the separation in the front end, reproducible chromatography, ion mobility when it's accessible, and the mass resolution. I think that's when we can finally capture the complexity as well as the dynamics range of the system we are look at, and then use a fragmentation library. So in waters we're thinking as a holistic way. I think some of the tools we have allows us to capture that complexity in multiple ways as well as the dynamic range. The technology, of course, we are focused on, and hopefully in the future we push the resolution and also the analytical peak capacity of the system. In the immediate terms, identification is a challenge, and we think we have taken a step forward with regards to that, with having some of the libraries now incorporated in our workflow. Methylene library, for example, is now incorporated in our workflow, and we're working on a number of collaborators to expand upon that, because we realize identification is the bottleneck. And also, the other interest is in going back to Vasilis was saying, MS imaging, right now we're looking at a global sample, like a serum, that's circulating sample, a urine. But now, I think next stage we're going to start asking the question of, we are exposed, where are these things that we exposed to being deposited? Is it going in the liver? Is it brain? Is it bones? Are they in teeth? So imaging will allow us to look at those things more in a spatial manner, and I think that's going to be another wave, and that we realize that, and we have some of that areas of emphasis as well. So for us, technology-wise, I think we're looking at it holistically. And data challenge is another thing that we recognize, because we are generating lots of data. And in one analytical run can be a very large file, a gigabyte of file. And one sample, one analysis. Now, how do we manage that? So we have some of the thinking behind that to manage that data to help the whole pipeline as well. So we have some thinking and solution from that end as well.
Thank you very much. So I would like to conclude. I think we had a fantastic symposium on the Exposome. I would really like to first thank Caroline Johnson, my co-chair of this, on this effort. I would really like to thank all of you guys participating. I mean, giving outstanding talks, and that was great for the students, was for the faculty, for everybody here at Yale campus, but also everybody attending the event. I would also like to thank our Yale School of Public Health for the support throughout this. But last and not least, this symposium would have not been made possible without Damaris. Where is Damaris? And Donna. So I asked my daughter Panorea to bring us some flowers for Damaris, and so I want to applaud Damaris and Donna. Thank you. I mean, you did the job. We just, so it's been a hard work to coordinate everything, trust me, and we might have missed a few things, but we apologize if we did. But I think at the end it was a great symposium, and we have a very good cheese and wine outside and cold cuts, so I would like to move the meeting now, although we're still going to be broadcasting. Caroline, do you have anything to say?
Just to follow up, I think we all need to thank Vasilis as well for putting this conference together. So a wonderful job.